Okay, so good afternoon, everyone. I'll have to start this in Portuguese and then I'll switch back to English. Boa tarde a todo mundo. Vamos começar então a defesa pública de tese de doutorado do meu aluno, né, Sérgio Novi Júnior. Bom, o Sérgio vai estar apresentando a defesa dele para a banca. Como um dos membros da banca não fala português, por opção nossa de convite, a gente vai estar fazendo a defesa toda em inglês e a gente teve a concordância de toda a banca para poder fazer isso, né? Então, com isso eu volto para o inglês. So, good afternoon, everyone. I'm back here in English again. So it's my pleasure to start the defense, the thesis defense, the Ph.D. thesis defense for Sérgio Novi Jr., who still is my PhD student over the past 5-6 years. And I'll have here the committee, which is formed by the external members, Professor Mamadou Diop from the Bang and Mine Institute at Western University in Canada. Professor Ana Osorio from the Mackenzie Presbyterian University here in São Paulo. Professor Mônica Alonso Kota from the Institute of Physics here at UNICAMP, and Professor Homi Zatux from the School of Electrical Engineering and Computing here also at UNICAMP. And committee is also presided by myself, who is the supervisor of the work. So we will do this in two phases. The first part of the defense, Sérgio will have around 15 minutes to present his work, and after that we'll go to the argution phase. Okay, so Sérgio, the floor is yours. Go ahead.
in which we're going to try to track these huge tracks in the brain. And you can also study the functional level. For functional, usually what you're gonna do, you're gonna try to see the brain, how the brain responds to a specific task, and compare the health group with the patient group. The nice part of the macro level is that you can access that with non-invasive neuroimaging techniques. And in my PhD thesis, I developed work in this part, in the macro level, particularly for the functional, the functional part, or for the functional neuroplasticity. So here we can, as I said, you can study both tasks and also the functional connectivity. For the functional connectivity, we're going to see how the different regions of the brain connect to each other. In this level here, for the functional, I believe that the functional near-infrared spectroscopy is a promising tool for several reasons. Most because the technique is portable and versatile. The portability and versatility of the technique allows us to study different populations at different scenarios. So, for example, we can study babies, and the baby is going to be set in the leg of their parents, so the environment is very comfortable, much different from an MRI scan, for example. We can also combine the FNIRS technology with these long fibers with the MRI, so you can perform simultaneous measurements of NIRS with other neuroimaging techniques, such as MRI and also EEG. And you can also take as the technique is portable, you can take the technique to the bedside of the patient and perform bedside monitoring. This is particularly very important for the intensive clinical unit. In intensive care unit, the patient, moving the patients is a little bit dangerous for the patient. So you don't have to do that. You can take the FNIRS and go to the bedside. So how the technique works? Just a quick overview. So as we can see in the figures here, we can place source and detectors on the scalp with the volunteers. So this light coming from the source is going to interact with the brain tissue and come back to the surface. Here we're going to, this is called functional near-infrared spectroscopy because we are working with near-infrared light. So this light is mainly absorbed by ox and deoxyhemoglobin. So by using two different wavelengths, we can measure concentration changes in both chromophores. So you can measure ox and deoxyhemoglobin, how they change over time with a high acquisition frequency in the order of 10 hertz. So why? But why are we measuring ox and deoxyhemoglobin? Why is this important? It's important because we know already that there is a neurovascular coupling in which brain activity, here, for example, I show the electrophysiological activity from the brain during a 4-pole stimulation in a rat. We know that this is going to induce hemodynamic changes. And these hemodynamic changes are well characterized by an increase in oxyhemoglobin in red here and a decrease in deoxyhemoglobin. So by placing optodes in the scalp with volunteers, we can access the hemodynamic changes, and you can later relate hemodynamic changes to brain activity. So we can measure brain functional activity with FNIRS. So knowing this part of the technology, so in this context, my work, my thesis that I'd like to defend today is that FNIRS is sensitive to functional plasticity at the intrasubject level in different experimental protocols. So I'm gonna do that. I'm gonna defend this thesis. I separate this thesis in three parts. In the first part, I'm going to investigate functional brain plasticity of infants at the group level. So we perform measurements in the brain of the infants, and you saw how it developed over time. Next, we're going to develop methods for block design experiments. So to better infer brain plasticity or to better infer brain response during a specific tasks. And we're also going to develop methods for resting state experiments. So I'm going to show you a lot of works that we have done. I'd like just to clarify that I didn't do this work alone by myself. I had a contribution from different institutes and different universities, such as the Hospital de Clínicas da UNICAMP, the State Federal University of Minas Gerais, Department of Electrical Engineering, Misericordia University, and we also collaborate with the Western University in Canada. So before I go into the work that I did, I would like just to give a quick overview of the current workflow for FNIRS experiments when I started my PhD. So basically, in typical Refinery experiments, we're going to put a lot of source and detector in the head of the volunteers with a cap. And to position this cap in the head, we're going to use a tape. Then we're going to set some landmarks in the school, position this cap, and perform these measurements. So the Refinery signals are characterized by, as we're measuring hemodynamic changes, we can see in the data, it has a heart rate oscillations. And to see if the channels are good or not, you have to measure at least the heart rate. So that's one way to see if the channel is good or if it's not good. So the current pipeline is basically, if the channel doesn't have heart rate, we are going to discard the channels because it has a low signal to noise ratio. Then, sorry?
Go ahead. It was just some mic open.
Okay. So then you can, for the remaining channels, you can estimate hemoglobin concentration changes. You can do some bandpass filtering, and then infer which channels are activated with a general linear model analysis. So by activated, I mean channels that had a characteristic hemodynamic response. So usually, since the signal to noise ratio from the technique is low, what you do is basically a block design experiment in which this volunteer is going to perform the same task different times. So for example, if you ask them to do a finger tapping, the volunteer is going to do the finger tap for, I don't know, 2 seconds. Then there's going to be a rest period, then finger tap again, rest, finger tap again. With this approach, we can try to model the hemodynamic response or the expected hemodynamic response with a general linear model, which basically, this is the design of the hemodynamic response expected for these blocks here. And we're gonna model the time series. Basically, we're gonna say that the measured signal, which can be oxyhemoglobin or deoxyhemoglobin, is going to be this model here, the hemodynamic response function, times a beta plus an inherent term. Then we're gonna do some statistical tests to see which betas are significant. So if you find a beta that's significant for both oxy and deoxyhemoglobin, we're gonna say that brain region or that channel is activated. Remember that a channel is a combination of a source and a detector. Another thing that I would like to bring attention here is that the light coming from the source and reaching the detector, it not only interact with the brain cortex, but it's also interact with another biological tissue that's a highly vascularized tissue, mainly the scalp. So hemodynamic changes in the scalp is going to affect our signal. So in order to solve this problem, we can use the so-called short channels, in which we're going to place a detector much closer to the source, and we're requiring this signal here in the short channel does not reach the brain. So we're just measuring the scalp or the extracerebral tissue. Then we can incorporate these measurements in our general linear model and perform the statistics from the GLM combined with the short channel regression. So this is the basic workflow for general linear models, GLMs. And remember that for a brain region or a channel to be activated, we're going to constrain the problem for ox and deoxyhemoglobin. So we should have a beta positive for oxyhemoglobin and it's statistically significant, and the same for deoxyhemoglobin, but the beta has to be negative. So with this approach, we investigated our first study we did in collaboration with the Federal University of Minas Gerais, in which we investigate how the brain of babies develops within the first year of life. So we perform measurements in baby of 6 and 12 months of age, use a probe here that covered most of the head, including the frontal, temporal, parietal, and the occipital lobe. In total, there were 84 channels here. In the figure, the sources are in red, in blue the detectors, and the channels are in yellow. And then we perform a sensory motor stimulation in the right hand of the baby with a vibrator. So our results are very interesting. In 6 months of age, we found that for the group analysis, we found the full-term babies, they had an hemodynamic response. In blue here, I showed the activated brain regions. So they had a broad activated region in the left hemisphere, which was expected because the stimulation was in the right hand. When they got to 12 months of age, the hemodynamic response became much more localized. So we believe that here for the group analysis, we observed a phenomenon of brain maturation in which the response in the brain became became more localized with as the baby aged. Then we also not only measure full-term infants, but we also measure preterm born infants. The results are very interesting. If you compare the preterm with full-term born, we see that at 12 months, the hemodynamic response of the preterm babies are much broader. We can see activated channels in the left hemisphere that was expected, but also in the right hemisphere that was not usual for an adult. And we can see that even in the left hemisphere, in which the contralateral brain hemisphere, we see that the hemodynamic response is much broader in comparison to the full-term babies. At 12 months, the hemodynamic response disappeared in the ipsilateral hemisphere. It became more localized in the left hemisphere, as expected. But if you compare with the full-term babies, we see that the hemodynamic response of the full-term is more localized. So we believe that the preterm born infant, it has a delayed maturation in comparison to the healthy infant, even after 1 year of life. This result was very interesting because with the Bailey tree metric, we didn't see any change between the full-term and preterm borns. Note that this was done for group analysis. So in the group analysis, we could see that the FNIRS technology is sensitive to brain plasticity. Then we asked ourselves, can you do better than group analysis? So I know that group analysis is very interesting and can tell us a lot of things about the brain of controls and also the patients, but it's also made for the group. So sometimes you have a group that doesn't have a response, such as here, in which I showed this is another study that I did not put my thesis, but we observed that for a group of infants, we do not see any response for the auditory test for the group. However, when we evaluated each volunteer, we saw that some of them had some specific brain response. So for the group, we do not see change, but some specific patients, some specific volunteers, they do have brain activity. So we thought, so can you do inter-subject analysis? Why is this important? So basically, when you're trying to learn something, it's better to know, it would be better to know how can you promote a better learning for each individual and not just for the group. If you're going to, for example, a rehabilitation protocol, it would be better to see which rehabilitation protocol is better for each volunteer. In order to do that, we're gonna have to access the brain of the patient or the volunteers several times. However, we have a problem here because the FNIRS inter and intra-subject reproducibility is very low. So, for example, if I measure someone today and then tomorrow, and I see some changes in the brain, I'm not going to be able to say if these changes are real or these are just fluctuations of the FNIRS signals. So what we try to do, which is the focus of part 2 and part 3 of my thesis, is how can you improve inter-subject reproducibility. So then we started by developing methods for block design experiments in order to increase the reproducibility of intra-subject level. So if you come back to the basic pipeline, we know that we have to discard the channels flow signal to noise ratio, estimate hemoglobin concentrate changes, bandpass filtering, and infer activated channels. One thing that you tried to improve in the pipeline was how to remove motion artifacts. We saw that it's a very important step. So basically, what are motion artifacts? To put the probes in the head of volunteers, we use several sources, several detectors, which are usually put for fiber optics. The problem is that when you have some relative movement between the optical probe and the head, is going to create some spool or some contamination in the signal, which is characterized by baseline changes, in which the signal is oscillating at some point, then it changes its mean, and also by some spikes. These artifacts here are not real, and if you do not correct them, we're going to see some change that not come from the brain, but just because of these motions. So the first thing we try to do is to improve the current state of the art methodology for removing motion artifacts. So the state of the art when you start this work was basically done by wavelet decomposition, and the wavelet is very good method, particularly for when you have abrupt oscillations the signal. So basically, this is an example of a time series. In gray is the raw data. So you see that you have some characteristic oscillations for heart rate. then we have a lot of movement here, and the signal is contaminated by the movement. When you apply the wavelet, it can remove these abrupt oscillations very well. However, it's not good when you have some baseline changes. So for example, here, again, here I don't have the abrupt oscillations, but I have the baseline change the signal, and you see that the wavelet is not good for correcting that because it's creating a trend in the signal. So we try to improve this by incorporating another motion artifact correction before the wave decomposition. So this was a spline. So in our approach, we did the following. First, we're going to do a spline interpolation and correct the baseline changes. After that, we do the wavelet decomposition. So it's a hybrid method in which we're going to do first spline, correct baseline changes, then you correct the remaining artifacts. So this is the performance of our model. It's much better for when you have the baseline changes. And when you have the abrupt oscillations, it doesn't change the effectiveness of the wave decomposition. So what we did then to evaluate our model, we acquired data from 42 volunteers in two tasks. The first one was the reading aloud, and another one is silent reading. Why is this important? Because when you are going, the regions responsible for languaging or for reading is the Broca region and the Wernicke. And above this region, there is the muscle responsible for moving the jaw. So when the volunteer is going to read aloud, is going to move their mouth, this is going to create some artifacts in the data. And these artifacts are very controlled. So we can use these artifacts due to the jaw movement to test our methods. So we did that. And I'd like to show here the group results for for this study. So I'd like you to focus on the B and C. Here, we can see that in the first line is the reading aloud. In the second line is the silent reading. These are the group responses for both tasks. In the B, I saw the wavelet performance. In the C, the hybrid method, which is our proposed method. So the idea is here, if you remove all motion artifacts, the reading aloud and the silent reading response should be the same. And you see that we have a better agreement between two tasks for our method, which is the hybrid method here. We see that for the right hemisphere, the agreement is much better than for the wavelet decomposition. So improve the current state of the art by combining an additional step before to the wavelet decomposition. These are our first improvement, the methodology. So we changed the pipeline, and now we added this new step, which are the motion artifact correction. Okay. So after improving the current pipeline for pre-processed definite data, we try to revisit the reproducibility of a very simple motor task, which was the finger tapping. So what we did was basically we acquired data within a week from 10 volunteers doing a finger tapping task. So the finger tap test was a block design experiment in which acquired data from in the first day, in a second day, in a third day, always at the same time. Our probe covered most of the frontal lobe and part of the parietal lobe. And what we observed is that for the first 5 volunteers, we performed the group analysis, and you observed that at the group analysis, the hemodynamic response from the right hand was well behaved and located in the left motor cortex, which is expected for adults, for healthy adults. So the result made a lot of sense. However, when you took the data from each single volunteer, we observed that, for example, the results changes a lot. And you would not expect changes in a finger tapping testing in health volunteers within a week. So these changes are not real. They are just fluctuations from the results of definite technology. So for example, here, in day 1, do not observe any single chain activated. In day 2, only one chain in the right hemisphere. Just in the third day, we found something that makes sense, and was expected. One thing that I'd like to bring attention here is that for this analysis at the intrasubject level, we did everything that was the state of the art. We acquired data at the same time to avoid fluctuations from the circadian cycle. We performed motion artifact correction. We did a GLM, the most golden standard GLM with short channels, pre-whitening in the robust fit, We combine the results from oxygen and deoxyhemoglobin. So for a channel to be activated, we should see a expected hemodynamic response for both oxygen and deoxy. And you also did additional measurements with short channels. So you did the state of the art. So we come back to our results, to our pipeline, and try to understand how could you improve this. So from where it's coming these fluctuations. So basically, we hypothesize that the fluctuations are coming from imprecise positioning and the reposition of the probe. So basically, I'm showing here some Monte Carlo simulations. I'm showing Monte Carlo simulations from the original probe, and the probe shifted by just 1 centimeter. We can see that this just 1 centimeter in the imprecise position of the probe changes a lot the sensitivity profile from the Monte Carlo. So we hypothesized that we should improve the probe positioning. So in order to do that, we developed in partnership with the engineering department from UNICAMP a neural navigator system in which we can use the MRI images from the volunteer in order to position the optical probe. So the idea here is basically, in the day before the experiment, we locate the motor cortex of each volunteer. Then we save that region in the system, in the navigator system. Then in the day of the measurements, we put the probe with a focus on the motor cortex to make sure that we are measuring the appropriate region, the region of interest. Then the first day, we save several points from the probe. We save several sources and detectors. Then the next days of measurements, we put again the probe at the same spot, and we guarantee the reproducibility in the order of millimeters with this navigator system, which is much more precise than the measuring tape. With this approach, we observed that the reproducibility increased a lot at the intra-subject level. So for example, here, if you focus on volunteer 2, what I'm showing here are the frequencies in which each channel is activated. So each ball here is activated channel, and the frequencies are defined by the colors. So 0 out of 3 means that the channel is not activated, then 1 out of 3, 2 out of 3, and 3 out of 3 sections. So here we can see that for the guided approach, in comparison with the standard approach, that's the measuring tape, we see that the reproducibility increased a lot. So we have a lot of channels in the motor cortex with the highest possible reproducibility. And you observe that from 5 volunteers, 4 out 5 volunteers presented channels in the motor cortex with the highest possible reproducibility. While from the standard approach, we just found 1 volunteer with a high reproducibility. And so here, if you come back to the pipeline, we made another change. So the first change was the motion artifact correction. The second change was the neural navigator system. So in order to get reproducible results, instead of using the measuring tape to place the optodes in the cap, we can use a neural navigator system, which is much more reproducible. We can guarantee a higher sensitivity to the brain, to the region of interest, and the higher reproducibility. So once we got these results, once you could make the inter-subject results reproducible, we try to apply these methods in a single volunteer. So I'm going to show you now a study case in which we try to infer brain plasticity before and after rehabilitation protocol. So this is a patient that suffered a damage, a head trauma when he was 28 years old. We measured this patient 2 years later the accident. So you can see here in the MRIs that the damage compromised most of his right brain hemisphere, in which he lost most of the sensorial and the motor functions from the left part of the body, including the upper and lower, the hands and the upper and lower limbs. So for this experiment, we performed Refinery measurements with a sensorial task, in which we placed a vibrator in the left hand of the volunteer, which was the damaged hand. We performed 1 measurement before the rehabilitation, 1 measurement after the rehabilitation. The rehabilitation protocol consists of 10 sessions of a combined the rehabilitation with transcranial direct current stimulation, TDCS, and a physical exercise with the typical rehabilitation protocol. So one thing that you observed that was very interesting is that at the first day of measurements, in the day before the rehabilitation, the hemodynamic response of the brain due to the vibrational task was located in the left hemisphere, which the ipsilateral hemisphere, which is the contralateral hemisphere from the expected hemodynamic response. So we believe that you observed here a change of function from the right hemisphere, which is damaged, to the left hemisphere. And here I'd like to highlight that the patient could feel the vibration of the vibrator when this was turned on. So he could sense it. And the brain region responsible for that changed from the right to the left. After the 10 sessions of rehabilitation, we observed that the hemodynamic response was still located in the left hemisphere, but it became much more smaller. So we believe that we observed here another phenomenon of brain plasticity, which was related to learning. This result is very similar to the one that we observed with the babies. So at this point here, we could demonstrate that the Refinery technology, it can be reproducible at the intra-subject level for block design experimental protocols, and we could demonstrate that it's sensible to brain plasticity at the single level. So one thing that we realized here is that when we're going for health participants, you can do activity tasks, for example, finger tapping, reading, and so on. When we're going for patients, it's very hard to make them to do the test. So for example, here, we're thinking about doing a finger tap, but the patient had a lesion in the brain, so he couldn't move his hand. So he had to focus with a sensorial task, with a passive task. So this brought your attention that you could try to go to other protocols. So for example, resting state, because in the resting state, you don't have to make the patient to do anything. So you can measure the patient while he's just resting. So what you try to do here, based on this experience, we try to move our development for studying the resting stage instead of the tasks. So then I go to the last part of my PhD thesis, which is the development of methods for resting state experiments. So basically, for the resting stage, you're not anymore interested on which brain regions activate during a task, but how the different brain regions connect to each other. So usually what you do, you measure the brain of volunteers from the whole head. Then you can try to take the signal from these regions. You are usually focused on low frequency oscillations and try to do some metric that compares the different response across the different channels. So here you can create a kind of correlation matrix in which each element I and J is a correlation of channel I with channel J. Then you can take a specific line from this correlation matrix and create the so-called functional resting state connectivity maps. So as I illustrate here, so you take one seed and you project the correlation of that seed with the whole brain. So this method has been shown to be very important for two reasons. First, healthy volunteers, they do have specific brain networks that are very reproducible across time. So this was shown basically for functional magnetic resonance imaging. So you saw that we can see that there are several resting state networks, among them the primary motor cortex, which is basically the correlation between the left and the right motor cortex. We can see other networks such as the FOMO network. It's very reproducible also for volunteers, health controls. And besides that, they can provide a lot of biomarkers for brain disorders, because several brain diseases such as Alzheimer's, epilepsy, and disorders of consciousness can damage these resting state networks. So if you are capable to measure resting state networks, we can do some longitudinal studies, because if the data is reproducible, and you can also try to find some biomarkers for brain diseases. So it has available in the clinic. And so, however, there is a few problems. When you try to do the same work as done here with fMRI, with FNIRS, you see three problems. The first one is that if you take a specific channel or a specific seed in the brain, we're going to see that this seed is correlated to all of the brain. We try to do this for several channels, and you saw the same. So, for example, here for sensory motor, everything is connected. For the auditory stage, everything is connected. So that's a problem, because the brain should not be connected with all parts of the brain all the time. The second problem is that the variability is very high at the intersubject level. So for example, here I'm showing the seats, the results for each volunteer. In the left is the group analysis, in the right is for each single volunteer. We can see that as I change the volunteer, I change the profile. These were all health volunteers. So if I see some changes here, I'm not going to be able to say if the change that you are seeing is because of some disorder or it's just fluctuations from the signal. Because even in health controls, we see a lot of variability. And the third problem is that the variability is really, really high at the intersubject level too. So what I'm showing here is the CD maps for one single volunteer across time. We can see that as the time varies, the profile of the correlation changes a lot. So that's the same problem. If I do several experiments with the same volunteer, I'm not gonna be able to say if those changes are real or if these are just fluctuations of the signal. So what we try to do is to solve these three problems here. So we hypothesize that this variability were not coming from real brain signals. It should be coming from some systemic physiological contamination. So what we tried to do was a controlled experiment in which we acquired data from 10 volunteers, all of them without any brain disorders, during 12 minutes of resting state. We employed a probe that covered most of the head with more than 100 channels and 8 short channels to regress physiological contamination. We also acquired the whole physiology from the patient, which includes the mean arterial pressure, the heart rate, and also the end-tidal CO2. So we had the whole profile of the FNIRS measurements with short channels, and we also acquired the extra physiology. Then what we tried to do was basically, we pre-processed the data from the FNIRS signal, each channel. We perform motion artifact correction. We estimate the hemoglobin concentration changes. We brain pass filter the signal in the region of interest, which is usual for resting state connectivity analysis. And then we focus on understanding this step here, which is how the removal of systemic physiology affects the Pearson correlation, or how it affects the maps. To do that, we use a general linear model. We try to use this model here because you can vary the regressors of the GLM. We change it from the short channels. We also combine we just using the model, the short channels for regression. Then we use the short channels combined with the other physiology, which is basically the short channels plus mean arterial pressure, heart rate, and end-tidal CO2. So with this model here, we can isolate how it systemically affects the data or affects the results. So the first result that I show is for group analysis, which I'm showing the correlation distribution. So you can see here, in gray is the correlation profile when you don't do any regression of physiology, in red when you do only the short channel regression, and in green when you combine short channel and extra physiology. We can see that all the correlation maps, the correlation distribution for oxyhemoglobin and total hemoglobin, they shift to the left, showing that a great part of the high correlations were coming from systemic physiology and not from real brain signals. This phenomenon occurs for oxyhemoglobin and for total hemoglobin because these two chromorphs are expected to be more contaminated by physiology than the oxyhemoglobin. For the group analysis or the seed-based analysis, we observe that I would like you to focus first in the first column here. So in the first column, what you see is the sensory motor map, seed map. In the first line, I doubt the regression. In the second line, only with the short channel regression. In the third line, the short channel plus physiology. We can see that as we're going to increase the regression, we're removing a lot of correlation. Then the maps become much more localized. In the end, you can see that it's very close to the expected fMRI results. So you can see also for the same behavior for auditory network and for the full mode network. So you can see that most of the correlation with the whole brain, the whole channels, they're not coming from the brain, but from systemic physiology. Then we ask ourselves, so okay, but acquiring all this data is highly demanding. Can you do it without acquiring extra physiology? So you try different preprocessing, which is based on the principal component analysis, in which you remove the first component with the source of highest source of covariance, and you can see that you can find similar results. So for the group analysis, we can try to use PCA instead of acquiring data, and it provides nice results with a good agreement with the fMRI literature. So next, we also try to see the behavior. This was for group analysis from 10 volunteers. We also saw the same behavior for every single volunteers. So each line here is a volunteer. Each column is a pre-processing. As we increase the complexity of the pre-processing, we start to get auditory networks that are much closer to the fMRI literature and much more compact, much more localized. And so the result was reproducible across volunteers for each volunteer in inter-subject level. Another thing that I tried to do was to quantify the reproducibility. So this is a measure of variability. So higher values means higher variability, lower values, higher reproducibility. So you can see here for the inter-subject level that when we remove systemic physiology, the results become much more reproducible. And we saw this behavior for the sensory motor network here in the left. We also saw for the auditory network and for the other networks that we investigated. And for the inter-subject level, as we do not have data acquired from different days, we split this time series of 12 minutes in 2 segments of 6 minutes. And you observe the same behavior. When you remove the physiology, the data become much more reproducible. Okay. So finally, what we tried to do was, okay, now that you have this data that is highly reproducible, can you try to do brain fingerprinting? So the problem of brain fingerprinting is basically, if I have a connectivity matrix from an unknown participant, can identify which participants that connectivity matrix coming from? So if you have a highly reproducible measurements, we should be able to identify volunteers, because the connectivity patterns from one single volunteers should be different from the others. So for testing this method, we did simultaneous measurements with FNIRS in the MRI. We did 6 runs for each volunteer for a total of 28 volunteers. And we tested 3 different metrics. We tested the geodesic distance, the Euclidean distance, and also a machine learning algorithm, which is basically a linear classifier. So we observed that, first of all, in order to extract reproducible results, you should have data from different runs from the same volunteer. So what I'm showing here is the average accuracy on identifying volunteers for the bold signal, for the oxyhemoglobin, deoxyhemoglobin, and total hemoglobin. With the 3 analyzed classifiers, we can see that for each signal and each classifier, as we increase the number of runs, we increase the capability to identify volunteers. So basically the data from each volunteers has some fluctuations, and some of these fluctuations are not just spurious, they are real. So in order to extract the profile from each volunteer, we have to acquire more data. That's the first result. The second result is that if you combine oxy and deoxyhemoglobin, these different chromorphers, we can improve a little bit the accuracy. It's not the same as acquiring more data, but it can help. And interesting, the combination of all chromorphers is not better than just combining total hemoglobin with deoxyhemoglobin, which means that oxyhemoglobin does not bring additional information to total hemoglobin. And finally, we try to verify how the number of ROIs impact the classifications. So we try to do the classification by changing the number of channels that you have. So basically try doing it only with 10 channels, 20 channels, 30, 40, and 48. And you observe that as we increase the information, the spatial information, we also increase the classification. So basically for extracting a good map or a signal highly reproducible for each volunteer, you should acquire data from a broader region or most of the brain. And finally, I would like to conclude this presentation or this work by saying the FNIRS is a powerful technique to investigate brain plasticity. So the main points that you found is basically FNIRS is capable to measure brain plasticity in infants with the group analysis. So you saw the brain evidence of brain maturation. for both the health and the full-term and preterm-born infants. We showed that FNIRS can provide evidence of brain reorganization after head trauma at the intrasubjective level. So you saw that you could improve the results of FNIRS at the intrasubjective level. Then you demonstrate that we are still capable to be brain plasticity, to see brain plasticity. The third point is that FNIRS-based resting state networks can be extracted at the intrasubjective level with a high reproducibility and consistency with the fMRI literature. So you saw that by removing systemic physiology, we can get reproducible results in the resting stage. These results can be used later for brain plasticity applications. And finally, our methodological improvements allow the extraction of individual features that could be used to identify subjects and ultimately to monitor brain changes due to functional plasticity at rest over time. So we have a really strong procedure methodology that can identify volunteers just based on their connectivity profile, showing that the intrasubjective variability is less than the intersubjective variability. Finally, I'd like to finalize this presentation by showing our pipeline, which is the main contribution of this thesis. So first of all, to acquire reproducible data, you need to place the optodes in the probe carefully. It's better to do with a neural navigator system than with just a measuring tape. After discarding bad channels, you have to remove motion artifacts. We advise you to use our procedure based on the spine interpolation and the wave decomposition. Then you can compute the hemoglobin concentration changes. You can do a bandpass filtering. And the most important is to do the removal of systemic physiology. The best approach would be to combine short channels with additional physiological measurements. But if you don't have that, you can use only short channels. It's an improvement. If you don't have neither the short channels nor the additional physiological measurements, you can do a PCI, at least for group analysis and intra-subject analysis. And finally, if you're going to do a resting stage analysis, you have to remove the autocorrelation of the signal, then compute the Pearson correlation. For blocks and experiment, you can do the GLM analysis to infer activated channels. So finally, I'd like to thank everyone that contributed to this work. First of all, Professor Rickson. Rickson has been my supervisor, not for the last 5 years, but for the last 10 years, since I was in the undergraduate, doing the physics course. I'd like to also thank all the members from the lab, especially Rodrigo Forte, which is with me since 2012. I want to thank Professor Tatiana, that helped me a lot with the statistical analysis. She gave me classes every week, basically. Also, Professor Gabriela for all the support and the advice. And I also like to thank all the group. It's a picture before the pandemic, in which you could take pictures without masks. And from this group, I'd like to personally highlight the importance of a few members here. For example, Giovanni Grisotti, that helped me a lot. And also Nicolas Gabriel. So thank you so much. And that's it.
Okay, thank you, Sergio. You were right on time because we started a little bit late. Okay, so now we go to the second part. I see that you were drinking a lot of water, so whenever you want to break to go to the bathroom, just let us know, okay? We can always close the camera and go to the restroom, but you can't. So let us know whenever you need it.
Thank you.
Okay, so starting with the second part, now we start arguing about the work from the committee, and then we can hear from everyone else that can have questions. So it's tradition here to start with kind of the furthest member. In this case, we don't have any questions who is the furthest member of the committee. So I'll actually start by thanking Mamadou Diop, Professor Mamadou Diop from the Department of Medical Biophysics at the University of Western University in London, Ontario, Canada. It's a great pleasure to have you on the committee, especially because we've known each other for such a long time. So it's also my pleasure to have you here and host you here in our as a committee member for this thesis. So Mamadou, Sergio is yours. You can question him for as long as you want.
Okay, so thanks, Rickson, and Sergio, thank you for the great talk. This was a huge body of work, and you did a fantastic job in summarizing everything in 15 minutes. So that was a tour de force. Congratulations for doing that.
Thank you.
So I really enjoy reading your thesis. It was long because it's a big body of work. And that's what you want for thesis. You want to hand to people something that they will feel that, yeah, this definitely deserve PhD. So it was a great you were before the talk you were talking about the English that was very well written. I think even for an English speaking person, this would have been judged as being very well written, let alone if English is not your first language. So congratulations for doing that, for you and your supervisors. Can you hear me better?
Yeah, I can hear better now.
Yeah. So now I have a few questions, as usual. So my first and foremost question is for you to go back and explain to me when you do the general linear model.
This one for the resting stage?
Yes. So this
okay.
One. So here, your measurement. Why is your measurement?
Why is the why is the hemoglobin concentration change? Okay, you can have oxyhemoglobin, deoxy, and total hemoglobin. So basically, this is for each channel. So you're gonna be model basically oxyhemoglobin equals to a linear combination of several regressors or several variables that could explain your data. So you are measuring physiology. What you cannot be explained by physiology, which is basically the residue of the model, is supposed to be from the brain. That's the idea.
Yeah, so if you could go to a whiteboard, maybe share your whiteboard and then start writing for me, that would be great because, yeah.
Okay, so for example here, suppose that you have the time series for oxyhemoglobin. Let's say, for example, oxyhemoglobin, you have the time series, right? So I'm gonna call this YHBO, right?
Okay.
So I'm gonna write YHBO is equals to, let's say here, the first measurements, let's say from end-tidal CO2, this one. Then we have here heart rate fluctuation, right? You have here. Then this is the near arterial pressure, right? And you can also have it's advisable to have a constant offset. It's like just once, for example, here, right?
Yeah.
Times beta plus the residual. So when you fit this data, you can do for a robust fit, for example, or just a least square fitting. We're gonna find these betas here that fit the model. This beta is going to be the best approach for find oxyhemoglobin, right? Just a linear fitting. Then you can write that your filtered data, y filtered, sorry, is going to be YHBO, which is the original data, minus X, which X is the whole matrix here.
Yeah.
Times beta. So this is going to be to filtered signal after removing, in which we're removing systemic physiology. That's the idea. And you can also include here the short channels, for example.
Yeah, so my question here is just the structure of the matrix doesn't fit for me because, yeah, you can have a, let's say an M by P, N by M matrix, which is X here. You multiply by a constant, that's fine. It's still an M by M matrix, but your Y is a column matrix. How do you handle it?
So yeah, this is basically we can do it should work actually. So this is Y, right? N by 1, right?
Yeah.
Equals to X. X should be N by M, right? Times beta. beta is m/1.
Okay, so beta is not, beta
It's a vector.
not, it's a matrix here.
Yeah, beta is a vector. Ah, yeah, sure. Yeah, beta is a vector.
Yeah, and how do you choose those beta values?
Yeah, the beta is going to come from the fitting, because you have this data, right? You have this data. So the beta is the fitting.
Okay.
Okay. So it's going to minimize the difference between minimize the residual, right?
Okay. That's clear now. That's clear. Excellent.
Okay.
Excellent. Okay, that was very good. So thanks, Sergio, for clarifying that, because that was
no
my
problem.
major question for the.
It is very confusing, actually.
Yeah. No. Now it's clear. Now it's clear.
Okay.
I could go back and revisit it and get a good understanding of how you do it. Okay. Great. So other than that, I'm just going to start with like general question.
Okay.
Okay. So for your probe positioning.
Yes.
Okay. So can you go back a little bit and explain to me how do you do it, like specifically? Because I saw that you have you talk when you're talking in your presentation, you talk a little bit about it, but I need a little bit more detail.
Okay.
Can you explain to me a little bit more detail?
Okay, sure. So the system is a combination of a hardware, which basically the Folhemos fast track. I use it to that. The Folhemos fast track, right? So it's basically a pen with a reference, so you can find a distance from the reference precisely. This is the hardware. The software, you have like we have an interface. It was developed by our university. So you need to have the MRI image for each volunteer. So if the MRI image, like we have some landmarkers in the head, right? So for example, here is a landmark and also the NZ and the union. So what you do is basically you take the MRI from the image, you locate this landmark is the MRI, then you put the probe in the head of the volunteers with the reference in the head. Then first thing you have to calibrate the system. So you basically is going to take each landmarker in the head, then you calibrate the system. So you're just going to do some rotation and translation to fit the MRI close in perspective to the reference, right? So then with the pen, you can navigate in the head of the volunteer. So the software, you can go with a pen here from the system, and you can see which anatomical structure is below the surface because of the MRI image. So in our study, use it all the time MRI image, but I know that you can also use other images, structural images, such as tomography, but I haven't done that. I have just used it with MRI.
Okay, great. Yeah, so it's good that you're thinking about that, because now there are these 3D topographic imaging where you just take a 3D holographic or structured illumination of a sample. So I haven't yet seen in human, but you can take you can have an object, just 3D, do a structured illumination. You can really have a rendering of a 3D image of the subject, and that could potentially help here if you don't have access to an MRI.
Yeah, this is going to be good, not for probing the probe, but for repositioning the probe. Yeah. Because even if you do like the 3D image of your head, you're not going to make sure the anatomical structure below. Even if you use the atlas, it changes a lot.
Okay.
So one idea in this direction, if I could just complement, is that you could try to take, for example, the profile of the scalp for each volunteer, then go to MRI base that we have a lot of them now, and try to get the volunteer that have the better match, the better scalp match. This could be an idea to decrease the variability.
Yeah. Okay, great. So you said that the maximum distance, and I think this was maybe, so yeah. So what is the maximum distance you can have between an FNIR signal and a source detector channel, while still being able to use that source detection channel as a regressor in your FNIR signal? So
Yeah.
just to rephrase it, so what is that minimum distance you can have between short channel and F-near channel while still being able to use the short channel as a regressor?
You mean the minimum distance or the maximum distance between the short and long channel?
Yeah, the maximum distance you can have between the two.
Between the two. For example, one short channel in the forehead and the one long channel in the back of the head.
Yeah.
Okay. So there's an open question. It's still an open question in the literature. So you precisely don't know that. But some recent works have shown that you should have it. The better way would be to have one short channel per source. And sometimes you could use one short channel per source and another one per detector. However, we're going to do now one study just to check that, how many short channels you should have. But one thing that I have done, I didn't put in my thesis and did not publish it, but if you allow some time lag between the hemodynamic, the short channel and the long channel, it improves the performance. So for example, you have some heterogeneity in the scalp, but sometimes it's just like phase shift. So for example, if you acquire data in the forehead and the back of the head, if you allow some phase shift between them, you could maybe use that.
Okay.
But for sure, I would say that it's better to have at least some short channels. Even it's better to have one short channel in the forehead than having nothing.
Okay, good.
Okay.
Excellent. Okay. So now on page...
The presentation or the thesis?
No, in the thesis.
Okay.
So you said on page 33 that frequency domain can also infer the time of flight of photon in tissue. So that was a little bit confusing to me. How can you use frequency domain F-nears to measure time of flight of photon in tissue?
Yeah, I think with the phase between the incident light and the light that's coming from tissue, if you have the phase, you know the average time that the photons took from going from the source to the detector, as far as I understand.
Okay, that is the right answer. Yeah, you take the phase difference that between your the phase shift that you have, and then you can use that to convert it into time of flight. Okay, good. So yeah, on page 35, you said that regarding the interaction between near-infrared light and biological tissue, an incident photon is absorbed when its energy matches energy differences in the rotational and vibrational mode of one of the molecules that constitute the medium. So the point I have there is that is in the infrared, in the near infrared, what do you have absorption like between fundamental mode, or do you have absorption between a mode and higher mode? What I mean there by overtones.
Can you say again just the last part of the question between
the? So in the near infrared region,
yeah,
is absorption due to just simple transition or due to overtones?
Yeah, honestly, not sure.
Okay, yeah, because fundamental there is a subtle difference there. It's not major, but in the near infrared you have vibrational and rotational mode. But in the so that's in the infrared. But in the near infrared, because the energies are wider, you have transition between energy levels that are not your delta n between your energy your transition is not equal to 1. It's 2, 3, or 4. It's a higher transition.
No, I see. Okay.
And that's why their probability is actually, if you look at them, their probability is actually smaller, although they have correspond to higher energy. So it's just a subtle difference there. Okay.
Okay.
Yeah, on page 36, you say the main molecules that absorb light in tissue are oxy and deoxyhemoglobin, basically blood. But don't forget water. I would suggest that you add water there because water is
Okay.
is a major absorber. You should not forget it.
Okay.
Okay, and just gonna move to, yeah, on page 94.
94?
Yeah.
Okay.
So you said that the high temporal resolution of FNIRS may lead to spurious correlation. Could you explain that?
Yeah, that's a good question. 94, right?
Yeah.
Yeah, about the autocorrelation. Okay, so when you are, for example, you have Mary waves, right? So it's about 0.1 hertz, let's say, or breathing. So when the signal starts to increase, you know that the signal is continuing to increase for a while. So you can predict the next point with some probability higher than handle. So the time series points are not independent, because once you see a Meyer wave, you're measuring several times, so you know that the next point the signal is going to increase. So for example, if you go back to my slide here, if I show this image here, can you guess where is the next point from the time series? For example, from this point here, can you guess where is the next point? We can. With such precision, of course, not infinite precision, but with such precision. So the time points are not independent. If the time points are not independent, you cannot use the Pearson correlation because it violates the assumptions of the model, the statistical test. So that's why this can create spurious correlation.
Okay.
Okay.
Okay, good.
And in the brain, like the signal I measure is much lower than the acquisition frequency. So this becomes a huge problem.
Yeah, so one thing I noticed there, just trying to choose here a few. Okay, which one? So I struggle to find. So in your brain in your brain fingerprinting study,
okay,
I really struggle to find the clinical applicability. Can you comment on that? How can you use this clinically when we have like very easy hand fingertip that you can use? Why would you need brain fingertyping?
Okay, yeah, this is a very, very good question, actually. So basically, you can use brain fingerprinting. One way of doing that is to develop methods. So for example, how do you evaluate? You don't know the ground truth, right? So we see the connectivity maps, but you don't know if that's one is better than the other, depending on some, you apply different pre-processing. How can you evaluate which result is better than the other? We don't know the answer. So one way that you could do that is decreasing intersubjective variability. And one way to evaluate that would be with brain fingerprinting. Because if you have a higher accuracy, that's basically because we have a higher reproducibility in the data. So these two things are aligned. So I think that the direct application is not in the clinic, but for developing methods. That will be the point. So for example, if you measure a patient today, and you then tomorrow, how do you know that the change that you are seeing is because the patient evolved or just fluctuations from the signal? So you should have some really good pre-processing in which you trust that to say, no, if I see change, that's because the patient changes, not just fluctuations. I would say that, actually.
Okay, so my last point here, this is just my last question, actually. I'm just gonna let you go. So in your last study, your subject, the sex of your subject was really skewed toward male. Why that? And is there any potential, confounding factor of that when you want to generalize the results or the conclusion you have from that study?
For the brain fingerprinting?
Yeah.
Yeah.
We have lots of male and very few women.
Yeah, that's one problem actually, because we're in the physics department. So our volunteers are our friends. So I invited my friends to do the studies, and most of them were males. That's why they are all males. Yeah, I know that's a limitation, but specifically for the brain fingerprinting, I think this is not a limitation. This may be a plus, because the cohort is much more less homogeneous, much more homogeneous. So this would be harder to separate a male from a. This would be easier to separate a male from a female, probably. Maybe, I don't know, actually. But if they are all males, like the very homogeneous group. But for sure, the study is limited by that.
Okay. Well, on my end, that's my last question. So I would like to congratulate you for answering the question very quickly and very concisely, and for the quality of your thesis. This is an impressive body of work. Congratulate you and your supervisor.
Thank you.
Thanks, Mamadou. Thank you very much. Very clever questions. And
yeah.
Yes, indeed, Sérgio answered most of them quite precisely. So now I want to pass the equation over to Professor Ana Osorio, which is the second furthest away. Right? So Professor Ana Osorio is a professor at the Center for Biological and Health Sciences at the Mackenzie Presbyterian University in São Paulo, a few kilometers away from us. And it's also my pleasure to host her here. And Ana, as you know, Sérgio, so feel free to question him as long as you want.
Excellent. Thank you so much, Rickson, for inviting me to be here today. It's a great pleasure. And I'd like to welcome you all, my colleagues here with me, and all the people that are right now watching this Viva or thesis defense. And I wish Sérgio that he goes on showcasing his remarkable work. Sérgio, I have just some comments and questions, and I wanted to ask if you would rather go on having this kind of back and forth conversation as you did with Professor Mamadou Diop, and maybe we can just have a nice conversation about your work, which was, and I agree with the previous reviewer or the previous member of this commission, that is quite impressive, very complex and interdisciplinary body of work. And I would like to start by remarking that and saying how excellent your work is. I've written down some things, not to get carried away. So before I start with the thesis itself, I'd like to make a very quick remark on your presentation, which was excellent as well. You very aptly summarized the main findings, the main ideas, the main contributions of your work, which was in a very clear and visually appealing way. So congrats on your presentation.
Thank you.
Regarding the thesis itself, it was a pleasure to read. I must confess, I'm not from the physics area at all. I'm actually a psychologist. I work in developmental neuroscience, but I'm a psychologist in training. So, Rickson, I have to be honest, when you invited me to be part of this Viva, I was a bit scared. I thought, how am I going to contribute in any way in a physics thesis? But I was very happily surprised that the way you wrote your thesis made it very understandable. Obviously, some very specific aspects of the physics calculations elude me, but it was a pleasure to read the work. And this is the mark of an academic, of a scientist, and I would say a very good scientist, is that you can deal and express very complex ideas in a very clear, concise, and compelling manner. So congrats on that as well. So I'll make some general comments, and then I'll just make some specific comments on specific parts of your work where I had questions, doubts, or I have suggestions. So your thesis is clearly innovative, and that's necessary for a PhD thesis. but it checks the box, obviously, but it also has very important interdisciplinary contributions. So when you think about all of the breadth of the scope of your thesis is very broad and very ambitious as well. So you had empirical studies with infants, with adults, and in those groups, either typically developing or healthy, typically developing in the case of infants, but also premature infants, healthy adults, and an adult who had suffered brain trauma. So studies based on functional protocols, but studies also focused on resting state data, development of a data analysis pipeline to reduce intra-subject variability in FNIRS experiments, an algorithm to correct motion artifacts, protocols for use of neuronavigation to guide probe positioning and reduce variability from external sources, custom adaptations of FNIRS systems. I could go on, but this is just a sample of all of the things you did, along with partners and colleagues, of course, but that you presented in your thesis. So as I said before, this is a very rich and complex piece of work, and I congratulate you and obviously also your supervisor who has some role in this, but mainly you, Sérgio, for this.
Thank you.
So, no worries. So considering my background and experience in neurodevelopmental science, I will obviously focus my comments and considerations mostly to chapters 3 and 4, which are the infant studies. I hope you can understand that.
Okay.
So I'm a prolific speaker. I'll swear, I'll just talk a little bit more about my impressions and I'll ask you the questions, okay? But I would just like to note that I was very interested and impressed by how you were able to articulate the relevance and usefulness of using FNIRS for the study of brain function and plasticity in young infants. And this is a relatively novel attention from neuroscience. This interest that is kind of recent on infancy is both welcome and long awaited, in my opinion. So with the contributions and interactions of different sciences, including physics, including psychology and other sciences, we can make this the decade of infant neuroscience. So infancy is a critical period of human development, and you state that very well, and you make that very clear in your introduction. And this, the fact that this is a critical period, coupled with significant advances in cognition, emotion, and behavior that we observe in children at this same period. So it's at the same time we can observe this behavioral explosion in terms of abilities that occur very quickly, but also we see a correlate trajectory in terms of brain development and brain maturation in terms of structure and function. So we are gradually uncovering the impressive functional and structural brain correlates of these changes that we see behaviorally. And this is a highly plastic and susceptible period, infancy, for better and for worse. And along these lines, you also mentioned in chapter 3 on page 50, how adverse early experiences, and I add, for instance, caregiver, psychopathology, neglect, poverty, may compromise optimal brain development. And in low middle income countries, which is the case of Brazil, as well as other countries, about 43% of children under 5 are at risk of failing to reach their developmental potential due to extreme poverty and stunting. And finally, on a brighter note, economic studies have also shown the higher return of investment in early interventions compared to later remedial interventions. So studying infancy and early childhood is a scientific and social imperative. And I've mentioned all this, thanks for your patience, because I would like to highlight how your thesis is not only original and innovative, scientifically speaking, but is also socially relevant as part of an effort to draw further scientific attention to infancy, thus contributing to moving it up the policy-making agenda. So this is my introduction, just to advocate for those who are listening, because Sergio already did his part, for all that are listening and may be interested in studying infancy from different perspectives. That please, you are very welcome. We need all hands on deck.
Yeah.
And so congratulations, Sérgio.
Thank you.
So I'll start my comments on page 49 on your thesis. So let me just find the comment. So we're on chapter 3, the first paper on infant development, where you compare premature infants with typically developing infants. And the title is Assessing Motor Function in 6-month-old Infants. And I would say this title doesn't really represent what you did in this chapter. So you assessed motor performance behaviorally using the Bailey scales. But the task that you selected for the FNIRS assessment seems to me more of a sensory task. So it's the mechanical stimulation of the palm of the hand of the infant. And as far as I could gather reading the description, the infants were not required to perform any motor action. They were just passively receiving stimulation in tactile fibers in their palms of their hands. Yeah. So my question is, if you had a chance, first of all, if you in any way agree with me, and if you do, if you have the chance to rethink the task, would you think of maybe a different task that might have assessed more specifically the functional brain responses to motor actions instead of this, like the sensory or tactile stimulation?
Yeah, so I totally agree with you. Yeah, I think that the title is probably a mistake, that because when we started doing this study, I thought this was motor. I was in the beginning of actually we started this when I was still in the undergraduate program. So I brought this mistake with me, but for sure it's a sensorial task. I agree with you. When you did this work, we did not know how to analyze open questions. So we just knew how to analyze close tests with specific duration. So now, when you ask a baby to perform a task, this should be an open task, because each trial, this baby is going to do the task in a different time. So, for example, if you ask the baby to get some object, for example, so it may be quicker in one time and slower in the other. So this is going to be an open question. So at that time, you do not know how to analyze this data, but now we do. So this kind of task of trying to involve the baby in some playing, some gaming, or taking some tests, some object would be better probably for isolating the motor task.
Like a grasping task, for instance. Yeah. Offering children toys and letting him or her have the opportunity to grasp the objects. Yes. Excellent. But you're right, it's very difficult because you can't control, you can have real block. Yes. Real blocks, because even if you present the stimuli in a block kind matter, infants, I would say you're not sure. Now, you're sure that infants will not pick them up at the same rhythm within the block. So that's a challenge indeed.
So, but now it's possible. You could do that. No.
Excellent. Then on page 50, you have the introduction on the next page. I just felt like it's a very nice introduction. You mentioned very important aspects. I just feel that there are some references lacking.
Yeah,
sure. So I can send you some suggestions, but several, important sentences and ideas that you convey need to be appropriately referenced. Okay, but that's something you can easily do.
Yeah, for this specific paragraph, this one of the last one that I did for sending the thesis, and I thought, no, in the end I could come back and put the reference, but I didn't. And I would like to just highlight here that I read your book for doing this paragraph, so you probably remember some. Yeah, I can get.
Well, it was easy for me because that's why I said I can give you the references, because I have them very, at hand. So I can give you, or you can read it on the chapter, of course. But that's very easy to correct. Yeah. No problem at all. So then, let me check. Then on page 51. You begin on page 50 talking about general neurodevelopment. and how early infancy is a very, critical period of brain development in terms of function and structure. Then on the following page, you talk about prematurity. And I felt that perhaps some mention of how prematurity or some detail on what we know is the effect or the implications of prematurity on brain structure and function. I think it would be interesting to have them here, have that information here. You mentioned it briefly in the discussion, but I think it would be worthwhile to mention it here. For instance, gross anatomical differences, so smaller brain volumes, difference in cortical thickness, differences in connectivity. But what's interesting is that there is some available data that shows that this is not linear. So, they're premature, so smaller brain sizes, less thickness, less connectivity, and it's not really exactly like that. So there's some evidence, for instance, showing that some sensory motor areas seem to mature faster in infants born premature in those first weeks compared to fetuses of the same gestational age. So something is happening and other areas of the sensory motor area of those infants lag behind in terms of maturation. So it's not exactly that they're just below or behind, but there are very qualitative differences as well. And it would be interesting to bring this here, this complexity here to the introduction.
Okay. Yeah. So, yeah, for me, brain plasticity is really hard, right? To fully understand everything. There are different volunteers, behave in different ways. So, I'm gonna try to improve here. So, but yes, I was very confused. Actually, when I was studying this, I was really surprised that damage in this early infants could be like to produce damage in much after in life, because the brain of these babies are really plastic. So at the beginning, I thought that they couldn't handle this, but there are some behavioral deficits and effects, right?
Yeah. Excellent. Then on page 52 on the method, and you describe participants. This is a question. I was just wondering, I'm not sure if you know this by heart, if you remember, were there any, so you say that preterm children included infants born with gestational age below 34 weeks. So you had moderately and very preterm, I imagine, infants up to below 32 weeks. But did you include any extremely preterm infants in your sample? So below 28 weeks gestational age? It's just I'm just interested.
Yeah.
Do you remember?
No, I'm not sure.
Okay.
Yeah, but I could ask the.
No, it's fine. I was just wondering. No worries. Okay. No worries. Just a quick note on page 54 on the figure 3.1, the B one. You say on the footnotes, you say photo of 6-month-old infant on its mother's lap. Sorry, it's a very simple thing, but I think you should change to his or her or their, but not its. Sorry, just a small thing. So on page, another question. This is more of a question to let you teach me why this is, more than I questioning you about this technical aspect. But on page 54 as well, you state the distances, the range of distances between source and detector in the infant cap, and it ranged from 1.9 to 3.5 centimeters. And I was wondering if 3.5 centimeters wasn't too wide apart for an infant head.
Yeah, you don't have to go for 3.5 because the distance between the scalp and the brain of the baby is smaller. It's not that big. But when you were doing the probe, we saw that there were one source from the detector, 3.5 centimeters apart. You just measuring signal with signal. So you kept that. So there is no reason for not acquiring that data. So we're still going to measure brain cortex. No problem with that. And the signal was good. So
okay.
But you did not plan to do a measurement with 3.5. But when you did the probe, there was some distance around that. and you decide to keep them instead of just throwing away good data.
Do you think the signal, because I imagine the answer, but I would rather hear it from you, which you are the expert in these specific aspects. Why do you think that the signal was good, even though the distance was very wide, even perhaps too wide for an adult, cap or probe or layout?
So when we are measuring the signal, we can see heart rate, the heart rate. So with the heart rate signature, we can infer that the signal is good from the signal to noise ratio perspective. So that
do you think this means that perhaps, because you said it was like incidental, you had that distance, you checked, the signal was good, you kept it. And I'm not an expert on the distances, because usually in FNIRS, the distance between source and detector in infant studies it's shorter than in adult studies. So it's counterintuitive to me, but I must admit that maybe something's missing for me in terms of knowledge about how, because we have these rules like, 2 centimeters apart is the more or less the ideal distance for infants, but then 3.5 worked.
So yeah, because the problem is basically, if I can jump in, if you go to longer distance, you are going to probe deeper in the brain, deeper in the head. Why is it easier in terms of signal quality to acquire good data from the babies? Because you don't have to go very far. The source don't have to go far from the detector, because they have a thinner structure in school and so on. That's better. So you can get good signals with this smaller distance, and you are still measuring brain. The shorter distance is better because we have more light coming to the detector. So it improves your signal to noise ratio. But it does not mean that with larger distance, you don't have signal. So in fact, we are going to probe deeper regions. So we are going to probe more brain. Your banana path, if you go back here, will not, here I extrapolated, right? So this is very far in the brain. But usually we're just measuring like very close to the most outer part of the brain cortex. So with a larger distance, we are going to be measuring a little bit deeper. Okay. So for this channel, 3.5, I cannot guarantee that
am I losing signal, or is Sergio?
Yeah, I lost signal. Is it me?
I don't know.
I think it's me. It says that my connection is unstable.
Yeah, I lost signal too, but I guess we're back.
Okay, yeah, back.
Okay, so you don't have to go here. Yeah.
I was just wondering because I'm obviously looking at the technique and see how this technique can suit my research interests, and thinking about, for instance, some
The problem is with them, because I can see you and hear you.
Okay, thank you.
Yeah, so I lost your signal.
Yeah, I guess we are all at the Institute of Physics, and apparently it's an instability at the Institute of Physics, because I guess both Sergio, me, and Monica, who's also here, lost the signal. But I guess we are all back.
Great. I was just wondering, Sergio, because I think about this from my perspective as well, because there are several areas of the cortex that we usually don't consider because they're a bit too deep. And I was just wondering, and perhaps born premature. you're born premature. So compared with a fetus that remains in the mother's womb, you face a different context in terms of temperature, pressure, gravity, textures around you, etc. But I'm also talking about something that it would be worthwhile thinking about, which is that we know that preterm neonates experience much more aversive touch, either painful touch, so they're pricked, they're to draw blood, to make all kinds of readings and measurements. They have a lot of invasive handling, which may not be painful, but it's invasive, it's unwelcome. During their early days, and even the more severe cases of prematurity, this could go on for weeks or even months. And we also know, behaviorally speaking, that preterm infants tend to be very reactive to tactile stimulation. They tend to be very easily bothered by tactile stimulation. So this is just a suggestion for the future, and I would just like to hear you think this is relevant in your opinion, in your experience, but it would be worthwhile exploring whether the frequency of such painful or invasive tactile experiences in prematures correlates with brain responses to your sensory motor stimulation task, for instance, perhaps coupled with another task more focused on a different area, perhaps the hairy skin, so not the palm, so that you can maybe stimulate a different kind of fibers, that like CT fibers. But I was just wondering what you think about this. Would this be an interesting avenue?
Yeah, I think that could be very interesting. But I also just wondering if how many subjects would you need to measure to do a good correlation to analyze this? We should have to. Do you think that a sample?
I think a sample such as this one would be, in theory, enough. Looking at not just the p-values, but also the effect sizes, and having some kind of measurement of, because everything is noted in these infants, reports. Blood drawn at this hour of the morning, then checking vital signs, then changing the thesis for the EKG, and so you have lots of moments, and everything is recorded.
So probably my collaborators in the Federal University, Minas Gerais, they do have this data, right? So I could try to check with them and maybe just complement the analysis. And yeah, that's a good point.
Because maybe that's more connected to the pattern of brain activation, the response to the tactile stimulation, more than the Bailey scales. So because I felt like the Bailey scales weren't very directly or immediately linked with the task that you used. So perhaps looking at another possible source of variation for the responses could be interesting. And I thought about this, the touch experiences that are notably very different between prematures and term babies.
Yeah, that could be very interesting. So I think I'm gonna check that, and maybe I can even send you an email later for.
No, that's fine. Don't worry.
Avenue.
Just a suggestion. Feel free to pursue it or not, as you feel it makes sense. Okay. On page 63. So just complementing, you mentioned that the Bailey scales of infant development that were used. Part of it was used to assess motor development. Might overestimate developmental status and be a poor predictor of later functioning, and that's in a way correct, but it is very difficult to assess development at very early ages, as you said. And one thing I just want to mention, and I'll just leave this out there and we can think about it. It doesn't mean anything, it doesn't require a response, but what we see early on in development is that the trajectories, even within typical development, are can be very different. You end up having a child that at age, for instance, 5 or 6 years is faring very well, but the rhythm and the trajectories are not the same. So heterogeneity is remarkable in early infancy, for sure at 6 months. So this heterogeneity, it really impacts the sensitivity and specificity of such scales, not just this one, but I mean, nearly all scales for this age. So maybe there's nothing inherently wrong with the scale, but maybe early typical development is really highly heterogeneous and strict thresholds are perhaps impossible to be defined. But this is just a comment, okay? Let me see this, we have discussed this already. On page 64, in the conclusion, you state, in summary, the results presented in this chapter suggest that full-term infants at 6 months have different task-induced activation responses in the motor cortex. My question is, can you really isolate the activation and say it was from the motor cortex, or perhaps it should be sensory motor area?
Yeah.
How sure can you be?
I think it should be sensory motor area, because even the SUCs probably not well defined to separate the prefrontal cortex. I think that should be sensory motor cortex.
Sergio, to be quick, and then now just moving on to chapter 4. So this is a very relevant work with a longitudinal design that is not often seen in developmental neuroscience for several reasons, but it's increasing, so excellent work. On page 66, at the last sentence, you say preterm infants respond differently to sensory motor stimulation, which agrees with the hypothesis that their brains did not have the same amount of time to develop as full-term infants. And I would also add, as I've said before, not only brain maturation at birth, but early tactile experiences are quite different between these two groups. So I would add some reference to, it's not just a matter of being born early and the system not being biologically mature, but then once they're born with that system, their experiences are also very adverse in terms of touch. So this, I think, is worthwhile mentioning. Can we go on? Yeah. On the results section on page 70, you detail the significant effects for the within group analysis. So you look at within the premature infants and you compare 6 months, 12 months, and you do the same for the term infants on the two most important measures that you mentioned here, at least. But were there between groups differences or group by age interactions tested? So like a mixed ANOVA. So because it looks like you look just at one group before, 6 months, 12 months, another group, but these interactions would be interesting to test. So perhaps in the future, looking at a mixed ANOVA design, I think, and perhaps, not for the thesis, don't worry about it. I'm just suggesting this so we can think, and perhaps in another opportunity, you can add something else, but having graphs that could visually show how for each of the variables, showing lines, one line for the premature, one line for the term infants, and over time, and showing really how these two groups are different or similar across time in these variables. I think above that, or more than just adding text, I think graphs could really quickly show us what the pattern was. Okay. And on page 73, you discuss one of the results in this comparison within group comparison, but I believe you did not discuss the result of an increase in the deoxyhemoglobin amplitude from 6 to 12 months in the premature group. I couldn't find it. Maybe I skipped it, but would you like to mention something about it?
Okay. I can add the...
Well, this is what I had to say about chapters 3 and 4. I'll make just some very general considerations about the rest of the thesis. On chapter 5, to be very honest, I have no technical expertise to appropriately assess this chapter and the novel algorithm you proposed, but I wish to congratulate you on developing what seems to me like a very useful and important tool for correcting motion artifacts. And this is, I was very interested in this chapter because you talk about movement artifacts, and as we both know very well, infants produce a lot of movement artifacts. And you mentioned the jaw movement. This was the main focus in terms of all different types of movement artifacts we could have. You focused on the jaw movement. And I was wondering about infants and how often they come with pacifiers or dummies, and they're sucking on the pacifiers. And I was wondering, what is your suggestion regarding use of pacifiers by infants during FNIRS tasks? Because when you remove them, you make the child fussy or fussier than they were before. So that's not good for task completion. And according to your results with adults, would you say that this algorithm may be useful in infants that we know are sucking pacifiers while doing the task? Or you say, or do you suggest, no way, it's better to remove and risk the child being fussy, not completing? Or otherwise you say, look, you can keep the pacifier as long as we correct this accordingly later.
Yeah, so for the motion artifact algorithm, I think that I did not explicitly say this in my thesis, but our original idea was to develop methods to remove motion artifacts from the baby signals, from the data collected for the babies. But it's really hard to do a control task for the babies.
Yes.
So we did it for the jaw movement. One idea to simulate an extreme environment, in an extreme situation, closer to the one for the babies. So this method, we did not validate it for the babies, but there was another work, was published just a few months before our work, that showed that the same method is really good for infants. So the method is validated. The paper is from Di Lorenzo, 2019, I think. It's a neuroimage. And they show that it's really helpful. So for your problem specifically, I would say that if you can remove from the baby, he behaves well, it's fine. If not, you can just keep that, because you can remove the motion artifact with this methodology here. So it's always better to avoid motion artifacts. But for babies, you cannot avoid that. So you have like this algorithm that's very robust and is already validated with baby data.
Excellent. And well, thank you for your explanation. And it's quite refreshing to see that something was originally thought for infants and applied to adults, and not the other way around, because it's usually always the other way around.
Yeah.
Things are adapted from adults, but this was the other way around. So congrats on that. And one final comment, Sergio, I'm just about done. It's actually, again, congratulating you. I thought that the last chapter and 10.1 on page 178, where you outline several guidelines for FNIRS data acquisition and analysis, is such a useful, interesting resource. And really thank you, because this kind of summary is very important for researchers out there that probably might not have the opportunity to read the entirety of your thesis. But I think these guidelines perhaps are worth publishing somewhere. I don't mean like a proper article, but as a resource for researchers. Okay. So congratulations on this. This is really a sign of generosity on your part, trying to summarize the main aspects that can help other researchers conduct better work with FNIRS. And that's it, Sergio. Congrats again.
It was so much.
Great honor to be here today. It was a pleasure reading your work, and wish you all the best for the future.
Thanks so much.
Thank you very much, Hannah. You touched a lot of very important points. I'll leave my comments to the end because I don't want to interfere with all the questions from all the committee members. But I'll just say one point, that when you mentioned about the background, right? And that was intentional. We really we knew that there was a lot. Sergio was touching very different topics with very different backgrounds. So we really wanted to get a diverse committee. We wanted to get someone from psychology that works with babies, and that's why we thought about you. We wanted someone with experience in years. That's why we actually thought about Mamadou. And then Rome is expert in time series analysis and data patterns and data analysis. And Monica thinks a lot about plasticity as well. So, I mean, we were actually missing someone from rehabilitation. We really wanted to have someone from rehabilitation background as well, but we could only invite 5 people, or 4 other people besides me to the committee. So that's why we are missing someone on rehabilitation, right? So, and in the end, I was actually checking, and we got a diverse committee, not only from the background, but also geographically and everything else, because we have Europeans, we have people from Africa/North America, and of course, people from South America here. So it was more diverse than we originally intended, and that is great. I really like the committee for this thesis. Okay, so we are halfway gone. Sergio, I guess I need to ask you if you want a 5-minute break to go to the restroom, drink some water.
Yeah, I think so. I think 5 minutes is welcome.
Okay.
Okay.
Okay, so maybe we can go back on in 5 minutes from now, around 5:10.
Okay.
5:14.
Excellent.
Right. Thank you very much. And then we start with Romis. You there, Holmes?
Yes, I'm
Okay.
here.
Okay. That's good. I guess. Okay.
Can you hear me?
Yeah.
Yes.
Okay. I
Okay.
had a problem with my computer's audio, so I had to connect via cell phone.
Oh, wow.
The audio, the video is from the computer, but the audio is from the cell phone.
Oh, wow. Okay, I'm glad it worked. Yeah.
Yeah.
Okay, so maybe I guess we can restart. So again, oh, Sergio went back. Okay, so now I'll invite Professor Gomes Atux from the School of Electrical Engineering and Computing here at UNICAMP in Campinas, Brazil. So Gomes knows a little bit about the project because he was part of the qualification committees. So Gomes, but now you have the whole thesis and the whole text and the final product, I would say, right? So my pleasure to have you here again. And Sérgio is yours.
Well, first of all, I'd like to thank you both for the invitation. It's an honor to be here. The work is extraordinary. I can only repeat the words of the previous examiners. The work is very well structured, it's well written, it's a very broad work. There's a lot of knowledge in this work and a lot of work in this work, a lot of effort in this work. And I think it was a sort of kind of tour de force, so to say, on the subject of news. So it was very pleasant to read the text, and it's one of these works that show that a research group as a whole is very mature. It really shows that your group as a whole is extremely, how can I say, extremely well versed in NIRS, in all aspects of NIRS, in experimental aspects, in theoretical aspects of the whole process. So it was a great pleasure to read the text, and I felt very happy about it because it was made in the university to which I belong. It was very nice to see that such high-level work is being produced in the institute. This is not something new for me because I have been in contact with the work of Professor Rickson's group and Professor Gabriela's group, but it always feels very good to read PhD work like this. It's really a thesis in the full meaning of the word. Well, so I have only a few comments to make regarding signal processing aspects and things like that. Let me take a look at my file. I'd like to begin in page 42, actually, 42. Yes. Right at the beginning of this page, equation 212, you mentioned that there are several options for the characteristic hemodynamic response function. We recommend using the gamma function since it performs well across several scenarios. Can you please talk a little bit about why the gamma function is generally recommended, and if there are, what are the other options you could have in this case?
So yeah, you have several options. So I can instead of giving the equation, I can draw some options. So the gamma function is basically this one, right? Sorry, I mean like so there is some others that some papers show that the hemodynamic response, there is an undershooting after the. So for example, this is the beginning. This would be the ending of the test. But there is some hemodynamic response that would be something like this. So you have this undershooting. So this is another option. So you can have a few equations that are a little bit similar to this one, but a little bit higher, faster in the beginning and slower in the end. There's some options, right? So when you are doing the experiment, what you're going to do is basically to convolve this hemodynamic response function to the block design of the experiment. So as I did in this figure here, right?
Mhm.
Here
Yes.
and here. So you are going to convolve them. So there is one paper from, it's from the Rupert group, one group from the United States, that does a lot of development of general linear models, and they check a variety of hemodynamic response in different tests. And they observed that the gamma function in general performs better for shorter periods and for longer periods.
Okay.
So even for shorter, I think that the gamma was the best one. And for longer, it was, I think, the second one, the second best one. And so we decided to stick with this one. It's the most simple. Yeah, that's basically the main reason.
Okay.
I think
that's
it's on
fine.
paper from 2019, I think, or 2018.
Okay. Okay. Next.
Yeah, this is the
Yes.
paper, 69. It's Santosa. It's 2019.
2009. Okay.
Right.
Well, then I have some questions regarding the linear model.
Yes.
You use equation 213, and you assume, which is quite usual, that the error is Gaussian.
Yes.
Well, and okay, you assume that is Gaussian, and later you perform the t-test
Yes.
for inferring whether the channel is significantly associated with the test. Is there a justification, statistical justification for using the t-test in this case?
You mean among other tests?
Yes,
Yeah.
yes.
So I think that the t-test is the most standard one. But I have seen other tests. I think I'm not really into the. I have seen other tests. I think the name is F-test too, but I'm not sure the details. But the fact is that we build this model for using t-test, but you cannot just apply the t-test because there are some violations of the model. So we have to correct for these violations. And one way to correct for them, I showed below here in the thesis, which is a noise violation when you do when you solve the linear model. So because, for example, you cannot have the autocorrelation in the residual. The timing points of the residual should not have autocorrelated points. And we have to solve for that. This one was the points.
Okay, all right.
Okay.
Okay.
But yeah, I know that there are other tests, but usually people do the t-test, and this is the most standard.
Okay, okay. In page 45, when you mention the noise violations to the Gauss-Markov theorem, right below equation 217,
okay,
you write here, according to the Gauss-Markov theorem, this solution is unbiased and has the smallest, the solution is unbiased and has the smallest variance of all unbiased estimator, if and only if. I think the way you wrote, you mentioned, according to the Gauss-Markov theorem, the solution is unbiased, but the fact that the solution is unbiased does not come from the Gauss-Markov theorem, comes from the properties of the solution, the 217 solution. So I would like to suggest that you break this sentence in two parts and mention the solution is unbiased, and it has the smallest variance. I believe that was what you had in mind when you wrote it, but I think that this might lead to some confusions regarding the Gauss-Markov theorem.
Okay, thanks so much for this correction.
It's no big deal, just a small suggestion. And then you have it has the smallest variance if the error term is not temporally autocorrelated, okay? And then you say that second part, it comes from a normal distribution with zero mean and constant variance. I'm not sure if the Gauss-Markov really requires that the distribution be normal. I'd like you to verify these. I think at the first point, the autocorrelation, okay, but I think it's possible to prove the theorem without resorting to the normal assumption. So I'd like you to, I think Gauss proved it first time, worked with this theorem the first time, considering that it was a normal distribution, but I believe Markov and more recent work managed to prove it without this Gaussian assumption.
Okay, I'm gonna check that because all papers that I read from the field, from the Refinery's field, they state that, but probably they are making mistakes, like one mistake after the other. Yeah.
Maybe it's a common hypothesis, but I do not work with this very much with these statistical aspects of least squares. I have only a general view, but I'd like you only to verify that.
Okay.
Okay.
So yeah, and just to complement this part here, when I didn't go into detail here because it was kind of a side problem, but when I do my solution, I don't do ordinary least square solution because in the near signal, you cannot guarantee the constant variance because of the motion artifacts. So we collect motion artifacts, but there may be some remaining motion artifacts. So the best solution currently is to do a robust fit, trying to ameliorate the outliers.
Okay.
But this is kind of the beginning of the.
All right.
Yeah,
all right.
The introduction for the problem.
Then you talk about pre-whitening, and you mention right below equation 219, page 46. You have for estimating the linear transformation W, we recommend the iterative algorithm by Barker, Arabi, and Huppert.
Yeah.
Why is this specific algorithm recommended, and are there other options, other possibilities?
So the first, there are two ways to deal with the colored noise. One of them is you have to assume some. You have to do some kind of pre-coloring, that you have to assume the spectrum of the frequency spectrum from the signal. And there is another one is the pre-whitening. So there is only one group with functional nears that developed methods for pre-whitening. This was first done in fMRI field and it was translated for the F-nears. This is the only group that do that. So in their paper, when they brought to the discussion, they state that there are 2 or 3 ways to do that, and they would recommend this way. And that's the only one that has been done in the F-nears. But I don't know if I should have removed the recommend and just say.
No, I just wondered if someone used the correlation matrix and some sort of decomposition of the correlation matrix. Even PCA that you mentioned later, perhaps could be used to perform this whitening stage. But I don't know. I remember from your qualifying exam that there were many connections between these models and source separation, and and this is really instigating. Really made me think, and I think I will talk to you later in the future to try to apply some techniques to this problem. But I have to study the problem a little bit further. So that just general questions, but later I believe I will look for you to discuss this point.
But here, just clarify, we're talking just about one single channel, right? This analysis performance. This analysis is all performed for each channel separately. Okay, so the PCA usually will use that for removing the correlation across channels.
Yes.
Okay.
Yes.
Yeah, but it's a very good point. So if you basically the idea here is that the noise in the data, I mean, here noise, there are different meanings of noise, right? So here noise, it means everything that's not the hemodynamic response. Yeah,
yes.
hemodynamic response. So like contamination from breathing would be a noise here in the low frequency. So if you would have like every single source of noise, you don't have a noise with autocorrelation, because this noise would be regressed. So, but that's not the case, right? You cannot account for all variability and all noise in the signal. Then this would be a white noise. So the source of noise here is physiological. The problem, right? The other noise, it's fine.
Okay. I think that perhaps you can use these different statistical properties of interference and noise and all these things. Perhaps they can be used to facilitate the separation process, the process of separating the hemodynamic response from what is not relevant. So, but I will think about it with more time, and I will talk to you. Okay, in page 79, it's exactly about this subject, the state-of-the-art motion correction algorithms, and you mentioned
79, 79, right?
79, sections 5.2. Yes, the second you have first you mention spline interpolation.
Yes.
And then you mention the possibility of using PCA and wavelets.
Yes.
And I think I only saw some moments before the beginning of this defense. I took a look at the Google Scholar for ICA and FNIRS. I think that ICA, independent component analysis, can be a valuable tool for solving this problem as well. So I think it's a point that you may look further in the future, in future work, in future efforts, you or the group, or when you continue to analyze this problem, because in a certain sense, it's more general than PCA. PCA is based on uncorrelationedness, and ICA is based on independence. So it's another point that may be interesting to analyze.
Yeah, I think so. If you are interested, you could try to do this in this work. We have tried to use ICA for another purpose. The problem with ICA usually is that once you do the decomposition, you have to look at the components and you say which components are noise and which components are not noise. And it's really hard to have an objective criteria. So that's people use that also for doing like instead of TD-based connectivity analysis, they use the ICA, sorry. So they're going to say, so they can find some networks, but the hardest part is to find the objective criteria.
Okay. Once again, I think that perhaps the statistical aspects of the artifacts in comparison with the hemodynamic response can allow this contrast to be established. So I think there's something there that can be explored, but it's a subject for future work of your group, your work and the work of your group. Okay, next we have page 82, right at the beginning of the page, when you mention the difference between Fourier transform and wavelet filtering. And you say, however, in wavelet filtering, the signal is decomposed into wavelets, which are functions that are well localized in both time and frequency. I think this might give a an impression to the reader that wavelet can perfectly solve the problem of having this duality between time and frequency, this uncertainty principle.
Yeah.
And they do not solve it completely. They are a different solution from Fourier. They have some properties that are interesting, but when you say they are well localized in both time and frequency, it seems that wavelets are able to solve this uncertainty principle, and they do not solve it perfectly. They provide another solution in comparison with the Fourier transform or the short time Fourier transform. So I think that it would be interesting to think about this sentence, these two sentences. However, and then you have note for comparison, the signal in Fourier decompositions decomposed in scenes.
Yeah, it should be localized at some extent or something.
Yes, yes.
Would be.
I believe you can emphasize that it's a
maybe
different.
Not emphasize. Yeah. Okay.
It has this time frequency character, and it's different, but it does not solve. It does not bypass the uncertainty principle that is inherent to time and frequency. Okay. Then you mention in page 131, 131, you mention ICA and PCA for pre-processing. And it's okay.
131.
131. Several pre-processing methods have been proposed to decontaminate. Among these methods, PCA seem to be the most effective, and these really attracted my attention. But for future perspectives, because I would like to see what kind of approach they are using for ICA, and if they are using the state-of-the-art methods, or if there's some room for improvement in ICA in the context of this problem. But I will look into it with more time.
Yeah, for this part here, for the ICA, I'm really interested in that, because it is another way for finding networks, and people argue that it's better than the city-based. So this is something that I'm really interested in doing.
Okay. Well, that's it. My comments were these. They are not essential. Only some reflections. Can you hear me?
Yeah, yes.
Okay. And I'd like to conclude by congratulating you and Professor Rickson for this work. It's a beautiful work, a real paradigmatic PhD work, dense and profound and broad. And I think that it says a lot about your competence and about Professor Rickson's competence and maturity as an advisor, and also about the competence of your research group.
Yeah.
So it was a pleasure to read it. I thank you very much. I would like to return the word to Professor Rickson.
Thank you. Thanks so much, Ramos.
Thank you, Ramos. Very important points. And some of them we are actually looking forward to discussing further, including the ICA. I must say, although this is not Sergio's work, but I must say that we tried ICA, but my idea with ICA was a little bit different. I thought that we could use ICA to find, instead of removing artifacts, to find the response, the brain response, just by automatically kind of removing its independence from all the other physiological noise, right? we tried it a little bit. It did work to some extent. It was not the best method we could find, but of course we are not even close to experts in ICA. We tried just general methods without really thinking about the parameters that we could actually twitch. So it would be very interesting to actually going back to this, because that's a paper we have it written, we never published because we have confidence on this, and maybe there's a better way to do it. Yeah, we definitely should talk about it. Okay, so I'll move to the last, or to the second last committee member, Professor Monica Kota, who's a professor of physics here at the Institute of Physics at UNICAMP, and my colleague here at the Institute. And so again, Monica, thank you very much for accepting the invitation to be part of this committee. You know that this invitation, it also comes because we spent hours and hours, literally hours and hours, discussing plasticity at the informal level over the past, I don't know, 5-10 years since I joined the department. So my pleasure to have you here. And Sérgio is here. It's all yours to discuss.
Thank you, Rickson. You're very kind. You know, when he described the diversity in the evaluation, the examining board, because I really don't know much about plasticity. My experience comes, well, whatever I know comes from, you know, personal experience. So, you know, Rickson knows we had a very, you know, very bad neurological problem in my family. And so I had to learn a bit, you know, when you're a researcher and you find a problem you cannot solve, then you try to grab all the tools you have around. And Rickson was one of the main tools for that. Said that, I have to acknowledge that, I'm not from the area, and everything I can, you know, say here is basically from admiration of the work that you've been doing. You know, Rickson and Sérgio as well. Sérgio is a very dear student here at physics. You know, I followed his work during those many years with Rickson, so I kind of, you know, got attached as well. And I have to say that, you know, reading the thesis, particularly because I'm not from your area, I work in material science, nanoscience, so it's completely unrelated, was a pleasure, you know, for many, many reasons, for seeing such a nice work, for seeing such a nice, well-written thesis, and the connection you made between all the steps you did, you know, showing very strong methodology in the analysis, everything you've done in your PhD, and I know it comes from your background in your research during the undergraduate studies as well. So it was really a pleasure to see how well, you know, a good researcher, excellent researcher you are now, Sérgio. So for me, it's, you know, being here in this institute for so many years, you know, because I'm really, really old here. It's been almost 30 years. So it's really a pleasure to see that we can have this kind of growing in our midst, you know, since you grew here. I remember when you were like in your first years of undergraduation. So it's really nice to see this accomplishment, you know, that you were able to develop. Well, so, but I have to make some comments and some questions. And so I'll just make some general comments at the end, but I'd like to ask you or point out some things I think haven't been already said, but I'll just point them out as well. So I noticed you kind of use your papers for the thesis. And when you use the papers, right, and you make a very, very brief, short summary at the beginning, right, of each chapter. But in the introduction that you wrote before, the first two chapters, they're not so, maybe so broad to cover the information that I missed, particularly because I'm not a specific audience that you were intended with your papers. I missed in the text. So I'll just give you a few examples. This is nothing that need to be corrected, but if you have the time, and some of them are already mentioned. For example, when you start in the beginning, page 32, and Oh, I have a more general question before that, not in the thesis. I always notice when you, in the several seminars I attended to from your group, you've put this kind of, oh, this is the signal as a function of time. So you have the oscillations due to heart rate, but also sometimes there's a low frequency noise. And, and your type 1 and type 2 artifacts, they're not there. So I was wondering, what is the low frequency noise? I think it's on your slide. I don't know if it's the thesis. The very first beginning.
I think, is this one?
No, before that. When you just say what's F nears.
Oh, here, right? This low frequency?
Yes. I've seen those a lot already. So you have this kind of, low noise that is not exactly low frequency noise. You have a high frequency in your type 1, and you have a step like in your type 2. So I was wondering how it relates. It's a type 1 something that you can, artifact that you can remove.
Okay, this one. So yeah, this one here. This one, it's a heart rate, right?
Yeah, that's the heart rate. But you see, there are like 2 bumps.
It's like this low frequency oscillation. No, it's not the artifact. This is real signal. This is physiological
signal.
It's real signal?
Okay, all right.
So the hardest part is to separate the real signal from the noise. So for example, here, in my example here, nobody noticed, but I'm gonna say this. So my wavelet here was probably too aggressive because I also removed the heart rate frequency.
Yeah.
So nowadays, later on, after this work, we improve the wavelet and it doesn't remove the heart rate.
Okay.
So this was one side effect.
All right. Okay. I thought you were looking at the artifacts in the first one. Sorry about that. Anyway, can you go back to the thesis?
Yes.
This was just curiosity. I always see this slide. So, okay. So when you talk about the, this is just an example, page 32 at the last paragraph.
Here, what? 32. Yeah, last paragraph?
Yeah, last paragraph. You see, you're talking about some general, the hemoglobin concentration changes and how it relates to brain activity. And you say, in the mature brain, which I assume it's, at least past teenage age, right? in an adult. But then you start to talk about the infants. And that's, I think, something Anna pointed out, that I miss what is the connection. So how you can take all these concepts and transport to the infant brain, which is really, really different, right? So a bit about the prematurity, what are the effects? And I understand that because this is already published and you probably use the paper, which is addressed to a very specific audience usually, or a more specific audience. So I think if you could add something in between these two, if you can use all the same concepts from a mature brain to make the analysis, or how the analysis changes, because you're looking at a very plastic brain for the child, for the infant, it might be maybe a nice attachment, a nice plus for your writing, for your text. Okay. Yeah. So there are a couple of examples, because when you mention the babies, you are just concerned about the signal. You say about the hair and the thickness of the skull. So your major concern is about the signal. But my question was how well you can translate the same concepts for the analysis.
Yeah,
for example.
Yeah. Go ahead. Particularly for the neurovascular coupling, this is very important point. So here, for example, I talk about the GLM in the beginning, the hemodynamic response function, and so on. But for the babies, it's not advisable to use the GLM because you don't know precisely how is the hemodynamic function from the baby. So this is something that I did poorly, try to address that with just a footnote, in which I say, here, do not use the GLM because the dynamics of the hemodynamic response may not be fully developed. But I know what you mean. I should expand this to give a more broader.
Yeah, but again, I understand that it comes from the format of you use in the thesis, where you based on papers. On a paper, you don't need to explain that probably, because whoever is reading is probably more in the field and knows a bit more about the differences. But if you make a more general approach for a general audience, it might be better to put that. Because, for example, the tonus in English of the muscles are different in infants. So even your motion artifacts will, you were talking about the pacifier, but your motion artifacts will be different because the way particularly at 6 months, 6 months is already, but a few months old baby is just really soft, right? It doesn't really have so much control. And that's something that, you might argue a little bit about. Okay. But something else about the babies that I didn't quite understand, or it wasn't completely clear, is the corrected age. When I read corrected age, I say, okay, so you assume if the baby was 6 months in belly and 6 months out of the belly, it's 12 months, or how you count the age? I don't know.
That's I think that's it. So you have to account for the weeks that the baby should be in the belly.
Okay, so the first time you say corrected age, just tell what corrected age is, because you mentioned in the end of the chapter how many weeks and so on since the last period of the mother, right? You say that, but you said that after maybe several pages and 2 corrected ages, which I marked down, and then I found out. So there are a few things that might be switched over. Okay. Another question that I didn't understand quite well, and it's again, it's not for you it's for you to enlighten me, because I really don't know. When in page 54, can you go ahead?
54, right?
54. Yeah. In the figure, yes. I noticed there you put the black lines to say the combinations that you used, so the signals you acquired, right? The pairs. Yes. But then you have LED connected to several photodetectors and photodetectors connected to several LEDs. How is this combination made during the measurement? I was curious.
So this combination. So for example, here, it's a good question. So the red one here is one source. This is another source. So AF7 is one source.
By the way, you should add it to the caption that the P, O, C, and F refers to the lobes, because it took me a while to find out.
Okay. I see. Okay. So
for you it's obvious. For me it's not.
So basically,
okay. How do you make the measurement?
So that's the idea. So you have A57 is one source, A53 is another source. So you should. And this one is a detector, right? The FP1. So if you turn on the light from both detectors, both sources, LEDs at the same time, you don't know the path of the light, because the detector is going to measure both of them. So what you have to do is you have to time multiplexing this in one round you turn on this source AF7, in another round you turn on the AF3. And this switching is going to define your acquisition frequency. So for example, for this measurement, I think it was 7.8, because we could turn on, for example, this source, we can turn on at the same time if this source in the back. Because the light coming from this source is not going to reach the detector in the front. So that's because it's too far away.
These are 2 wavelengths, right?
Yeah, each LED is 2 wavelengths.
Okay. Oh, each LED has 2 wavelengths. This is the double. Oh, okay. All right. So you have then 2 wavelengths in 2 different positions, and you multiplex the signal that is arriving into the photodetector.
Yes.
Okay. So that puts a limit on the frequency you can make the measurement, right?
Yes.
Okay. Thank you. It's a dumb question, but I wasn't sure.
No, it's very important.
Okay. So if you go ahead a little bit and page 58. Yeah, this figure, yeah. So you have all these additional channels. You mentioned that you have all these additional channels with which are not at sensory motor cortex, and you see some activation. Are these all physiological changes? Because in that work you haven't used all the methodology that developed after that, right?
So this can be physiological changes, or can also be brain activity. I think it's hard to isolate that at these ages.
So could you use the PCA there, for example, to try to clean it up?
Yeah, that's a good point. So people are doing PCA now for babies. There are some people doing PCA. One thing that you'd like to do is to try to use the PCA and compare the PCA with the physiological measurement that you have. So trying to find how can you combine the data from the whole head to extract the physiology. That's very important. So people are trying to do this right now. That's the next thing.
Okay, all right. So because there's a lot of things. Actually, in that curiosity, the people from Minas Gerais continue working with the babies. So did they have any further information? I think Anna asked something about, but I missed the answer. So do you have any further information on how these babies develop, for example, for laterality? Because of if you look at the preterm, you had some activation on the right lobe, the other lobe that shouldn't be active, right, at the time. Remember when you see the activation?
Yes.
So did you follow up on these babies to see how they develop? And one question in particular is laterality. I think that's the name, lateralidade in Portuguese, where you have the right hand or left hand, and how they ended up, as not a baby, but a child, a toddler. Do you know if they followed up?
Yeah, I know that for the second time, much fewer babies came back to perform the measurements at 12 months. So I think that they may have some they may be tracking some babies, but I think that most of them don't come back don't show up again. But I can ask Dan again. So we have done other studies with Dan, but for this specific study we kind of stopped it. But I can check that. That's a good point. We can try.
If they have any even if it's not at years, but if they have any data on how this baby is developed at I don't know how many years it has been that since the measurement.
Yeah, they should be 7 years old now. 6 years old.
Wow, that's a lot. Yeah.
Yeah.
And something else that was in my mind on the discussion on this work is, of course, you see a difference in the group, right? Between the preterm and the full term. But can I say it is the development is not as fast, or can we think about if the preterm are more plastic than the full term, so they can find ways more easily, not so focused? Because once you get this more defined area, that means you're getting out of the plastic period, right? Because if the plastic, you have a lot of synapses. I may be saying something completely wrong here, so forgive me, Anna, or whoever is speaking. But can we think about that it's more plastic because it needs to be more plastic to compensate for not having the full development in the belly? Is there any paper that discusses that?
Yeah, that's a very interesting question. I don't know. I haven't thought about that. It's the other way around, right? So.
Yeah, I just don't know what is the current status in this area. So, but if I look at it, I can look the other way around. So they keep plasticity open for more time to compensate for. Yeah.
It's basically the brain is more plastic in the beginning of the life. As the brain matures, it loses plasticity, at least a part of that. So if they are less matured, they may have higher plasticity. That's the rationale, right?
Yeah.
But I don't know. I don't know. I do have to check for that. I don't know.
I don't know. It's just an idea that came because it's really hard to talk about this because I don't have any experience with babies other than my own and a few other from friends, but every baby has a different development, even if it's completely full term, if it has any damage or brain damage or anything like that. So sometimes, and it's also really, really interesting because I remember that once it started to talk, it stopped to walk. Right. So once it starts to develop a skill, sometimes another skill which is already being developed kind of goes to the background because something is taking over. So it's really interesting from the point of view, of course, I'm not a neuroscientist, but it's really interesting that there are so many differences in the development of the infant. And I believe it depends also, of course, on the stimulation and on the environment that it's being, the child, the infant is growing up. But maybe you could, Anna is saying something here. Yeah, plasticity or immaturity. Maybe it's it. Anna can probably help out with this question, but it's just a thought that came to me. If it's a tool that you, for example, when you have, in my case, I saw something like that in my family. But when you have, for example, people, girls who have any kind of problem in development, the period might be delayed, because they need to, the change in the body will demand energy, and if they don't have energy, they cannot do that. So there are kind of adaptation mechanisms to overcome the barriers that is being imposed. So I was just thinking about if it could be the other way around, but I probably, Anna, can provide any more thoughts about it than I do, and maybe you do as well as physicists.
Yeah, no, also.
Okay, so can you go to figure 5.6? It's a big jump.
5.6.
Yeah, I think everybody's tired. Just again, just curiosity. It's page 92.
This one?
Yeah. So remind me, you have the Broca's area in B and C vernix area, right? And you see this difference. Yeah, if you look at the red and the magenta curve, you see this difference in the profile, right? And vernix area is related to speech. So, but the Broca area doesn't show any difference. I was wondering why, because they're both related to speech, right?
So the difference here, I come from probably remaining motion artifacts.
Oh, okay.
So, yeah, so that's the problem. So probably there are more remaining motion artifacts in the Wernicke. But one thing that you could like to bring attention here is that there is one previous work that showed the motion artifact induce one hemodynamic response that's about one order higher than the motion artifact induce one order higher hemodynamic response. So here we emphasize that the hemodynamic response, they do look different at some extent, dynamics, but the amplitude is not that far, that's not that different. Just to emphasize that you could remove at least a great part of the motion artifact, but some of them probably
but the shape of the responses is still affected.
Yeah, it's still affected,
right? Because
that's
they're quite
the reason.
different.
Yeah.
Okay. Yeah, I was wondering why they're so different. Yeah, that's something else that I try to find in you talk about the systemic physiology effects a lot, right? It's part of your work as well. But I had a hard time finding out what they are. I mean, of course, somewhere in the beginning, I think on page 43, you mentioned blood pressure change, respiration, heart rates. In page 99, you talk about circadian circles. So it would be nice if you just define in the beginning what kind of physiological, effects you could have and how they affect and which ones you're referring to. So, for example, I'd probably be a really bad subject patient for FPers because I have this kind of adrenaline reaction, which you probably can see I'm red every time I talk or I get red, So my students usually say, "Oh, Monica is red, go away."
No.
But that kind of physiological response would be probably very bad for FPs,
Yeah, so that's the problem actually. How to separate these changes that are correlated to the task. Suppose that change is speaking. If you get red, so there is something well correlated to the test. So how can you remove that?
Yeah,
that's the problem.
Yeah, in that sense, for example, for the group of patients that you used, of course, in the last chapter, you control some variables independently by adding the extra measurements. But in the beginning, it should be nice to say what kind of physiological change you are considering or you're addressing, right? So to make it more clear to the reader.
You mean each chapter, or do I do this in the?
No, maybe in the beginning. First time you mentioned, which I found systemic physiology effects. I think it was page 43 in the introduction chapters. Okay. I'm not sure if you mentioned it before, but I used Control F to find out because I was missing that. Okay. And you were adding more information in each chapter, and I was kind of, wow, I'm lost here. What kind of systemic physiology changes are usually addressed when you removing this background? For example, for me, you probably have to make to find a baseline when I'm already red, right? So because otherwise you get the ramp, it won't work. Anyway, there's something else that I think it's missing. For example, on page 141. But you don't need to go there, so I can just mention.
Okay.
The references, you mentioned that, and that's something that I really don't like, Sergio, to be honest. You say, oh, this makes sense. Oh, this was expected, right?
Oh, really?
And you know that brain science is not exactly, expectations are not always fulfilled, right? Because everything is changing, the dynamic of the new knowledge is arriving. So something that was expected a decade or two ago are not expected anymore.
Yeah.
Knowledge change. So in the text, you mentioned was expected according to fMRI measurements, right? And there were no references there in at least two places where you can Google, you can find it over the text, but there were no references in the text. So when you say something was expected and not to be confused with the fake science, oh, I'm seeing this because this was expected. You know what I mean?
Yeah, I know. I understand.
Take the data. Oh, this is expected, so this is right.
Create the hypothesis after seeing the data.
Exactly.
Yeah.
But just to make sure. So in the text you mentioned, this was expected according to these measurements or according to this interpretation, according to this paper. But there are no references there. So either it's very common knowledge, which for me, again, I can't tell, but I think you could add some references, right? And I'm finishing here because I know you're tired. General questions. Oh, by the way, I love your English, as I mentioned, but in page 175, you made such a nice comment. I have it here, 175 in the end. You say chapters 3 and 4 beautifully complement each other by revealing insightful aspects of early human. I love that.
I was a little bit afraid of putting the beautiful.
No, but that in that place it was okay. It was just the conclusion perspectives, but I thought it was really, kind of poetic, in a sense. So I like it. I even put a star on the outside.
Yeah, I was afraid. Some friend, I showed this to some friends. They told me, "Oh, I think that's fine, but I wouldn't put that." Then I said, "Oh, what should I do?" Then I kept.
No, that's okay. That's your style. It is kind of a leave of absence from the very hard and technical text. But I think it's in this place. It's your conclusion, so it's your emotion coming over. I don't think it's bad. No, I liked it. Anyway, something more general. In the conclusions, you talk about the low cost, high portability, ease of use of these techniques, right? Which might allow daily individual target patient treatment at his or her own environment. Well, I know if you compare to MRI, you're right? But I can't really tell going with all those fibers and that. I think it takes like an hour or so to put that thing in the right place. Yeah. So how about the future? If you could go can you do this in a more, I don't know, less invasive way, in the sense that you know, have to cover the head with all these things and have these cables hanging on? You know, it reminds me of the rat, the mice that has the fiber on, the optogenetic stuff. And I always look at it and say, oh, poor guy has this thing hanging on his head all the time. So the patient would be in the same situation. So what is the future for this? If you really want to make this, go to someone to just put something in your head. How do you call it? The toca in English?
Cap.
Oh, cap, yeah. Okay, so you put the cap on your head like you're going to a swimming pool or something like that, and then you press a button and it shows all the nearest signs. Is it possible? Is there's any future in that for let's say?
So yeah, the future is the wearable technology, and the people are trying to come up with very nice solutions. And some of them, you just put the cap and you move around the optodes, and it takes away the hair. So the current problem is probably one of the big problems, the hair. But there are some solutions in this direction, but we're far from that. But for sure, the technology is growing fast. And for measurements in the forehead here, like it should be very, very easy for the new technologies. Like so the technology is evolving. So the technology is going.
How do you see that happening in
Yeah, so I think it's going to take a few more years to be really. So I think that this is one of the state of the art. So Gower Labs. So I can put a cap, then you position each module in the head. So there is not so many fibers that we have in our system.
How do you carry the signal from?
So there is one cable here. Can you see here? It's very small.
No, I can see.
Yeah, there is one cable here.
Okay,
so
the cable is connected.
The cable is connected. So that this is the nearest equipment is basically here, the small box. So this is one of the new technologies. So yeah, the technology is going for wearable designs.
So you can move the optodes or around for the area that you want to probe?
Yeah, you can move them, but you have to do it before the experiment, right? So you define some positions. Then you just go with you have this one here holding the system with the cables for the electrical energy, right? Then you come to another module and you plug in.
Oh, that's nice. Okay, so
but the problem is
feasible.
The hair. Yeah. Where there is hair, there is still a problem. You're gonna have to remove the hair. It's gonna take time.
Yeah.
But the technology is evolving very, very hard, very, very fast. For babies, this system was specifically developed for infant studies. The main goal.
Oh, that's nice. Good.
This one example.
Okay. Yeah, I know that. You know, it all depends on material science, right?
Yeah, it depends on material science. Thanks for the hard work.
Well, material science is a mixture of physics, chemistry, engineering. So it all comes back to physics. Yeah. So that's exactly why I would ask, because if you have neuroplasticity, or if you want to probe any I don't know, any research on neuroplastic behavior of the brain, so on, using FNIRS, you show that the spatial localization is important to remove the artifacts, because you use the neural navigation and you get better results, right? And of course, maybe you cannot use neural navigation, but you know that precision can be helpful. So when you have neuroplastic behavior, you don't exactly know what is going to happen. Like in that case of the trauma that he studied, now the guy used the other hemisphere to control, right? You have cases in the literature where people have only one hemisphere. They're born with one hemisphere.
Yes.
And they can pretty much have a normal life because the plastic behavior helps. But can you, I think the picture you showed answered this question, but can you, for example, have a tunable position? So you can, for example, let's say you made a rough measurement. I'm just thinking as a material scientist, maybe. You screen your sample, right? So you get the rough measurement of what's going on, and then you focus on one area or one effect that you want to probe. So can you configure your cap to do that? So for example, you just do a rough measurement and get all the electrodes and then make a general analysis, and you see there's an activation area, like you in the trauma patient, right? That is maybe expanding or growing, but you don't know where it is. You have to find out from this first experiment, then you can make a configuration specific to probe that area with more detail, like the microscope, for example.
It's a very, very good idea, actually. I haven't thought about that before, actually, but I think that's a really good idea, because you can decide the position of the optodes. So for example, for that case study, in the second measurement, I could probably take all the source detectors from the right hemisphere and put everything in the left hemisphere, so it increases my sensitivity to the brain and map better the regions. I think that's a good idea. I haven't thought about that before.
Yeah, but maybe this new technology with the variables, you might be able to do that, right? And do some fast measurement just to get the probe, or where is activating here for this kind of the tickling with the feather that they did. It was not a feather. I think it was a do you call it cotonete? Forgot. Anyway, that might help, because you really don't know what is the nervous tissue that is being used for that specific kind of measurement, right?
Yeah, but for our assistant, we can also do something similar. It may take more time, but you mean like right in the first measurement, right? You do the first measurement quickly, then you focus. Yeah, this could be possible.
Yeah, because you might be able to increase a little bit the resolution of your measurement if you have a larger density of detectors and sources, right? I don't know. So I was thinking what kind of new measurements, because it seems to me it takes a lot of time for you to make the analysis of the signal. So I don't know how feasible is to do that, also from the time frames involved.
Yeah.
Right. So if you do the measurement and it takes like 3 hours to get, this is the activation area, or 1 day to get the activation area, you cannot come back and just rearrange the setup. Right. So that was also included in the question. Is it feasible from that point of view?
So depending on the protocol that you are going to do, it's hard. It's really quickly, really fast to analyze the data, because if the block designed and everything is fine, like a typical experiment, we can do really quick now with the method that we already developed. We can analyze it very fast. So the hardest part is going to be to put the montage in the head, and also to think about a new montage faster for doing it the same day. So this could take a few hours just to optimize the montage. But I think that's an idea that we could invest some time on that. I think it's worth trying.
Yeah, because I think something that I was reading, it's a completely different issue, but I was reading a comment in Nature yesterday. Yes. And it was about validation, validation in the sense, it was about a gender issue and so on. But they were saying that sometimes people just drop off because of validation. You don't feel that you're doing a good work because people don't acknowledge that, right? We are very fast in pointing out the problems and making criticisms, negative criticism, saying, this is wrong. And so I'm talking about my students sometimes, but we're not as good in doing, in praising that the work is well done or in things like that. So in that sense, that makes a lot of difference in the lives of many people. But I was thinking about, in that sense, the fact that you have a feedback, right, in your plasticity.
Yeah.
So that also would help you, maybe, because you have to realize neurological rehabilitation is really an effort, because the people always get more tired than normal. If you do an exercise with a neurological impairment, it takes a lot of strength and a lot of will to do that. So if you could make something that maybe give you a time frame, for example, if you see this, the trauma patient, he could evolve. I'm not talking about placebo, nocebo effects, okay? But it maybe get the sense, not because, I'm feeling better, no, my brain is focusing better. And that may help these guys to continue in this path, right? Or to also to make something that for me was quite difficult, is how to assess if the rehabilitation protocol is okay.
Yeah.
Because as brain development in child and in everybody is different, the rehabilitation protocol, you mentioned that the interpretation is also different. But the therapists make a lot of difference, right?
Yes.
And that was my main ordeal, is trying to be sure that the therapist knew what she was doing, because I heard a lot about, oh, it's a neurological patient. We don't know how far, this person will go. So you don't know what is the limit. So you assume the limit is the person, is the patient. But
yes.
I saw that a lot of times the limit was a therapist.
Yeah. So yeah, that's one of the main goals of this work, like trying to infer if a treatment or if the procedure is doing well, if it's the most appropriate. So maybe we're gonna see like, no, this rehabilitation protocol is not working for this specific person. Let's change for this another one. Let's change for the other one until we find the best approach. So in order to speed the recovery and go farther.
Yeah, but as I mentioned, it's a tool for the therapist to evaluate her own work, because that's a little bit a complicated situation, because the evolution of the patient, the therapist regards, the evolution of the patient is this, because the patient is being limited by their impairment in the brain. But sometimes just the protocol or whatever the therapist is doing is not well, even if it's the right protocol.
Yeah.
That's my point. Even if the protocol is right, but the therapist training is not good. And unfortunately, we've seen a lot of medical doctors giving chloroquine to the patients, right? And saying that they are cured because chloroquine cured them because, they didn't die of COVID. So we know that the evidence-based health treatments, for example, is really lacking, it's really missing in a lot of places, particularly here. So it will be nice not only to say, this protocol is better for this patient and so on, but also to assess if the choice made by the therapist is, and how they proceed. So it's a bit more than, I think, just choosing the right protocol. Is also choosing the right professional. The
right combination of protocol and professional. Right?
Exactly. Okay. So I think you have more to praise about your work than you really said. Okay. Sérgio, sorry for all these, questions that are just generic. And again, I love this subject, but I really can't contribute much. But I want to, again, acknowledge it's a really nice work,
Thank you.
I think it's really, really, really nice. And I'm particularly proud of the researcher you are, right? And I know Hickson has contributed a lot for that.
Sure, yeah.
And so I'm really proud of Hickson being our professor here in our faculty. But I always like the diversity in that you brought here together in this meeting. Because, for example, Mamadou, I'm sorry if I mispronounce.
Yeah, it's Mamadou.
He caught the question about man, which I think I missed. I saw there, I compare man and woman, but I missed, and I'm really kind of gender aware. So I think it was really nice and shows how diversity is important, particularly at this kind of scenario we're dealing with. And I appreciate the comment, Mamadou. But unfortunately, it's true. A lot of the girls, we have a lot of women in undergrad physics here, enrolled in physics undergrad studies. But as they graduate, sometimes they move away. So we lose a lot of our female students. And I think it's because of the way we handle the course and how there are a lot of questions involved. But that's why Sérgio has more male students around him than female, unfortunately. But I mean, we want to change that, right, Sérgio?
Yeah,
I
sure.
think so.
Of course.
Okay. So again, thanks for the invitation. It is a pleasure. It's a very nice work. For as somebody mentioned, it shows how the group evolved since Rickson started this line of research here, and I'm really, really glad that I was able to share this moment with you. Thank you.
Thanks so much, Monica. Thanks so much.
Thank you, Monica, and thank you, everyone. Very interesting, very insightful comments. We, for sure, I took notes, a lot of them, and I also recorded the whole section, so we can actually go back and recheck whatever we missed. And I know that Sergio is taking notes as well. All right. So thank you very much, everyone. I'm not gonna question Sergio, because I guess that's what I've been doing for the past 10 years. And about this work specifically for the past 5 years. So I guess I've questioned him quite a lot over the years. But I do have some comments that you guys did over the questioning that I was taking notes. So maybe I want to go. I didn't want to interrupt because he is the one being evaluated, not me. So I wanted him to deal with the questions, right? Which he actually did quite well. And from, I didn't have much to add, but just a few questions, I guess, just a few comments, right? And then I guess I'll start backwards and I'll start from Monica. I guess you touched one point that it's something that we've been concerned a lot, which is kind of the research near system but the difference between the research near system and the kind of practical NIRS device that can be used. And I guess we are reaching to a point, and mainly in the NIRS field, that really we can think of a neuroscience of every day. And that's kind of what has been driving a lot of interesting research questions. I guess Anna touched on this a little bit when she talked. But also it's like we have companies, Sergio showed Gower Lab Systems, we have Kernel, which is also starting. But if you go to their website, that's their goal, to make real life, everyday neuroscience systems, right? And when we do this research, I mean, we are interested in kind of digging really deep and really trying to understand the questions and trying to understand what makes a difference, what doesn't make the difference. So we are always meticulous about kind of the experimental details we do. And particularly Sergio, I guess, if there's something that he learned very well, it's this very being meticulous about every single detail that matters. So he was really good over the years. He became very good in being very kind of detailed about what he's doing and trying to get the best signal. If he has to spend 1 hour to get one single source detector well and good, he will do it, right? And that's not what we typically do when we are thinking about neuroscience application or everyday neuroscience applications, right? So all the kind of experimental details that it's in this thesis, it's always thinking about what's the best thing that we can do. And that's a different constraint from everyday life, right? And the same applies for data analysis. We try to do everything offline, and we take sometimes quite a while, and Anna knows that from the data that we collaborate together. But it really, because we try to really think about every single detail and every single step, every single parameter. By changing it, how does that change the data and things like that, right? So that's not necessarily a constraint that you have when you think about everyday systems, right? So I guess talking a little bit about the work and a little bit about the details, and I guess I'll start with the babies studies, right? So I guess maybe the first comment that I want to make is actually the data started 90 years ago, right? They started, Sergio actually traveled to Minas Gerais with me for the first time. I guess it was late 2012 or early 2013. I don't quite remember. But it was really kind of almost 10 years ago, and the field developed a lot over the past 10 years. Because Mamadou can tell that, and also can say that. And so that was a really, it's what, I guess it's documented. It's mostly kind of our progress over time, even with the data analysis, right? So chapters 3 and 4 are what we taught about back in 2016, 2017, when these first papers were actually, when the first data was analyzed, mostly because we didn't know what to do with some of these motion artifacts from the babies and things like that. And some questions still remain. So you guys touched on a very important one. How is the baby brain and how does the baby brain works, right? And the honest answer, we don't know, right? One thing that actually we need to, and I tried our best to acknowledge our limited knowledge, right? So, but we learned a lot over the past years. We know that for sure, the brain is not, when the brain is not mature, it does behave differently, and we now have some kind of suspicious candidates to why, but it's not published yet, and that's an experiment that we wanted to do in the next few months. But every single kind of advance, it needs another experimental protocol, another baby, another cohort, and things like that. So it's, it can be very slow, right? So about this cohort that it's published and it's in the thesis, right? So all the babies are in the kind of, at Minas Gerais, they have this program that aims to follow the babies. And it's a prematurity, it's not only a prematurity, it's also a social vulnerability program. So they actually try to track the babies up to the 7-8 years old, because it's kind of the age, the scholar, the age that they actually start going to school. And I guess most of what we know from psychology is that these babies are delayed in the cognition part, and they have consequences on the school age. And that's where most of the clinical data is known. And as Anna put it, we don't know much about the early stages. And that's why we start collecting data at the early stages, because we hypothesize that this can be, we can have early biomarkers that can show this, right? So to answer the question, yes, we do have data from these babies up to 7 years. We don't, they do. But yeah, they do collect clinical data. NIRS data, we just have up to 2 years old. After that, we didn't collect more data. But yeah, we do have a plan. And for the kids that actually developed ADHD, we collect more data than when they start at actually 6, 7, or 8 years. So we do have data from a sub-cohort of this data. From the premature babies, we didn't get extreme prematurity. So all the babies are actually in between 29 and 34 weeks. And the reason was most of the babies who actually were born before 28 weeks, they do have a lot of the exclusion criteria because they have a lot of problems, and they need ventilatory support most of the times and things like that. So they just decided to exclude these patients. They do have a study with all these extreme babies, but it was not part of the collaborative studies that we were doing with them, right? The corrected age is related exactly to this difference. So because the babies, the premature babies were born before time. So you have a few weeks of difference compared to the full-term babies. So that's why, and therefore, if you want to compare them at 6 months, right? They're not exactly at 6 months postnatally, right? So that's why you have to correct the age to make sure that these premature babies, you are comparing the premature babies at the correct ages. that the full terms are. So that's what the corrected age meant and that thing, right? Anna raised a very good point. Well, two very good points. One that I completely agree. It's not a motor task. It's a sensory task, right? And I guess we should be talking about the sensory motor cortex and not the motor cortex. Even the changes that we see are kind of more anterior and more towards the sensory motor compared to the motor tasks. So that's correct. And that's something that we should fix in the thesis and should be quite straightforward. The correlation of the Bailey tree is quite poor. And as you said, there's a lot of questions about the Bailey tree scales, right? One thing that it's worth mentioning, and we didn't actually put this on the not even in the paper, but I'm not an expert about the Bailey tree, but I guess it has a lot of different domains, a lot of different parts. And there's one part that's specifically to the motor task. And there you see a little bit of more correlation compared to the whole Bailey tree test, which is the test that they were doing. But we didn't know how to explain that. And we didn't really understand, and we didn't even kind of trust of the correlation because it was kind of very mild. So I guess that somehow that got lost over the years and over the analysis. But yes, that was something that we were trying later on, trying to look at the different aspects of the Bailey tree and the different domains that the Bailey tree corrects. Although I completely believe, and I completely agree with you, it does make sense to look more at the touch and the tactile experiment, because that's definitely a kind of more straightforward correlate of what we are measuring than the Bailey function, right? And so yeah, so that was something related to the experiments with the babies, right? One thing that actually you questioned and made me think about it is whether it would be worthwhile trying to do a motor task instead of a sensory task. I guess Sergio answered quite nicely too, and that's something that it does make sense that the complacency, right? The compliance of the babies to perform the task, and there was a methodological issue at that time as well, as Sergio explained it. I guess there's another reason, which is the motor cortex tends to maturate before the sensory cortex and even the auditory cortex. So I think that if we really wanted to do something at the motor cortex, we should be doing this before the 6 months age. Otherwise, we probably are at risk of all seeing the matured brain. So that could be something to think about, right? Another point that I guess it was Anna who raised as well, the most frequent against the average channels, the average response. And that is something that I guess Sergio really struggled with, both in terms of thinking about it and proposing a solution, and removing that or having that out of his thesis, because that's something that he spent a lot of time thinking about it and trying to work out a solution. And I guess we did think of a solution, and I guess we really need to think about neuroscience as a field in terms of mostly cognitive neuroscience, because most people, 90% of the studies, people really think about, oh, let's think about these two groups and let's compare the average of the group averages against each other, right? And it does have a place, but behind this, there are a lot of assumptions. And one of the main assumptions is that the system is ergodic, right? And you think of humans as balls, right, in kind of physical systems, right? And you think that two balls are basically the same, therefore you can just interchange them, and the group average is the same, and all the fluctuations are just random fluctuations, which is not the case for human subjects, right? And if you really think about it, well, first of all, every member of your group will never be identical. That's one reason. And that's per se enough not to do group average only, right? And the second thing is actually, they will not remain the same in the future, and that's what motivated Sérgio's thesis at some level, right? So yeah, we've been advocating for a few years now about not looking at the average response, because in a homogeneous group, and it's a natural homogeneous, heterogeneous group, because the subjects are not the same, they're not just two balls that should be the same, right? It doesn't make much sense, right? You have a very different response from kind of just as a normal curve. So we don't have a nice solution. I guess the best solution we've been proposing quite a while is to look at the single subject response, right? That's what people should be, that's what I think that people should be really thinking about it, and not just the group response. But I guess even if you want to look at the group to get more power and to say something and to reach some conclusions, I guess there are better ways to do it. And I guess the better way that we found is to present a frequency response, because at least you say something that most of the responses or most of the subjects within that group will have. Right? So that's what we've been doing. The problem is that we don't have statistical validity to show that this is indeed better, and that's what Sérgio has been working with Tatiana from the statistics department here at UNICAMP, so that we can actually have a kind of theoretical background to support what we've been seeing experimentally and empirically saying, right? One thing that actually I want to point out, I don't remember who actually talked about it, it's about the artifacts, right? So, and there's always this question about removing the motion artifacts or correcting them, right? And I guess Sérgio really said what we all believe, in the sense that it's always better to avoid, right? And therefore not have the motion artifacts. In some studies, that's just impossible, right? He brought up one study here, which is studied with babies, right? The only way you can do that is if they are sleeping, which sometimes is not what you want. And but even we do a lot of studies in the physical, with the physical exercise department, when people have to move, right? And there will be natural motion artifacts there, no matter what. So in this case, you need to correct artifacts, and you need to find a robust way to do it. And I guess we found one good way to do it, which proved to be very robust across different experiments we've tried. Although that doesn't, it's better than, we can say that it's better than what we compared with, right? We did not compare with ICA, and that may be a room there. But we did compare with the kind of most traditional approach in the field, which is called this CSPI, contrast-based signal, right? And it turns out that it's the worst method that you can find. Ours is kind of at least 3 or 4-fold better, not only in terms of improving, but in terms of not removing not losing the channels that are with artifacts, right? Someone pointed about the guidelines that we had in that he had in the conclusions, right? In the chapter 10, and I guess the idea, and he was, and I really wanted to say that because it was his idea, not mine. He really wanted to have an opinion paper based on this, right? So the original idea is to write an opinion paper about about kind of current guidelines that we could use with all the variations of neural experiments. And I guess over the years, he has acquired a lot of knowledge and empirical knowledge that what really works, what really doesn't work. So, but the problem is actually we didn't have enough time, and he has something aligned with a deadline. So that's why we didn't come up with this paper, but that's still on the table, and we hope to really write this paper on analysis guidelines soon, but it won't be in the thesis. So, but that was the original idea. That was the seed that originated the section 10.1, right? I guess I already mentioned about the ICA. I feel like that's a powerful technique. We really felt that. We really wanted this to a different aspect, to really identify what we want instead of just identifying what we don't want, right? And but yeah, maybe we hit a wall that we can't go further, but definitely we are interested in going back and revisiting this problem. Sérgio worked on this as well. It's not in his thesis, but Sérgio, one student of mine before Sérgio, Rodrigo, worked on these, and then Sérgio followed up on him, and we didn't reach what we really wanted to, and that's why we stopped it. Maybe it's time to get back to this. All right. One point that Mamadou brought, and it's very interesting. It's about the neural navigation system, the comparison with the 3D topographic image, right? And I guess Sérgio explained what we could do with the 3D tomographic image, which I agree. But I guess what I would compliment on Sérgio's response is actually, there's a very tiny difference with the neural navigation system that for us, it makes all the difference. And that's the possibility of you identifying where you are before you start the experiment, not after. So with most of the 3D topographic images and all the kind of registration techniques that we have currently in the field, they're all concerned about making sure to know where your probes are with respect to the head or to the scalp, but there's not really what you can do once you know where the probes are, right? And I guess the beauty of the neural navigation system that he designed with with colleagues at the engineering school is that we can navigate, we can do something before we start the experiment. So we can really navigate and put the optode where we want before we start the experiment. It's not just a passive response. We can actively put the probe in place, right, where we want. And that was originally not designed for a better registration or even for Sérgio's work. What we really wanted to do with this neural navigation system is measurements at the ICU. Because when we go to the ICU, right, well, we know that most of the times we want to measure patients that has a very kind of local brain injury, right? And we don't really know where it is. We can put the probe, we can register that, but it would be really nice to know where you are and put the probe exactly in the injury or in the penumbra around the injury that you have. And that's really what we wanted the neural navigation system for. And but then it turned out that we found a different use as well. And so again, and basically it turned out that we can use that to also improve the stability of the probe, and therefore the reproducibility of the results in a longitudinal study as well. But that was not the original aspect that we wanted it for. We really wanted it for to go to one very single spot, kind of the hemorrhage that the patient had or the stroke that the patient had, and navigate there, make sure where the stroke is and put the probe in between the probe. So I still believe that that's the best approach because, and going back to what Monica was telling in the end, right? If you want to do rehabilitation, maybe that's what you want to do, right? So that you want to really monitor one specific point or the region around that point and things like that. So if you can navigate before the experiment was done and make sure that you are on the right spot before, maybe you don't even need 10, 12 channels. Maybe 1, 2 channels would be enough to cover if you know exactly that you'll be there for sure, where you are for sure, right? I guess those are the main points. All the other questions or I guess Sergio answered, and I don't have anything to add, or they're actually very good and excellent ideas that we should definitely incorporate in the final version of the thesis. So in the end, I guess most of you talked about the thesis and the work that was done and the translation, or even the translation from the paper to the thesis. And I just want to remark, yes, I guess part of the reason it's well written is because most of this text was already reviewed and published, right? So we actually, there were a lot of people who were reading different parts, but not the whole thesis, as the committee had the opportunity to read. But we did change a little bit in the introduction from the papers to try to address this problem. But yeah, I do agree that there are some texts that maybe after a while in the field, you are so used to it that you don't even try to explain. So it's good that you actually raised some of these points that we can go back and add a few sentences or a few references that can help, right? And I guess just to end my comments, right, I just need to say that, yes, Monica mentioned, right, what a pleasure to see Sergio here as in the defense. And I would say that for me, it's really both not only professional, but also personal pleasure to see Sergio defending his thesis. And I don't want to say much at this point, but over 10 years, it's more than just one student. We developed a good friendship and someone that really we can talk to, not only about work, but also personal stuff. And I guess he specifically went through a lot of things, a lot of ups and downs, a lot of changes over the past 10 years. And it was really, to me, as not only as a supervisor, but also as a friend, to see how that comes out in terms of professional work. I don't need to say much, but I'll tell you that when he actually approached me back in 2012, so 10 years ago exactly, and he was just a second year undergraduate student in physics. He didn't speak a single word in English, right? So, and I remember that, I guess 2013, when we had the visitor here that was a native from native English speaker from the US, and we went to the bar, and Sergio always tries to speak a lot, and as you could see over the defense, he was completely mute because he couldn't not only speak a single word in English, but he couldn't understand a single word in English, right? So 10 years later, to see him presenting his thesis in English, I'm sure it's a great accomplishment for him, but it's also an honor for me to see that and know that I was part of this back in 2014 when he decided to go abroad and study English, which really changed his English level. So yeah, there's just one single example. There are many, but I guess we are already running out of time, so I won't say much about this. So yeah, it's a pleasure and it's an honor. I guess at some point I read a sentence, right, that if you as a professor, as a mentor, are not having students that are better than yourself, there's something wrong, right? So I guess to me that's enough, because at least I have the peace in mind that I guess I did something that was not quite wrong, although I'm sure we have a lot of mistakes on the way. Right. So with that, I guess it's time for the committee to get together and deliberate about Sergio. The way I'll do this, or I'll at least attempt to do this, is to create breakout room, and I'll try to invite committee members. I'll just have to make sure you can do this.
Português.
Yeah. Ok. Então, de volta ao português, que eu preciso dar o retorno em português. Sérgio, a banca, o comitê, sentou, avaliou a parte de arguição e a parte da sua apresentação e decidiu que você está aprovado como, com título de doutorado. Em ciências, não é em física. Então, parabéns.
Em ciências, não. Obrigado.
Enfim, se você quiser falar alguma coisa em português, inglês, your turn.
Yeah, I'm gonna speak in English just so that Mamadou can also understand. So I just want to say that I'm gonna take Rickson again. So thanks so much, Rickson, for all the support over the last 10 years. Yeah, I have a lot of things to thank you. And I know that the journey was very long and it was hard also because of the work. It's hard, right? So, but in the end, everything was fine and we did a great job. And I'm really proud of the work that you have done. And I also want to thank Professor Monica because she signed my papers for going to Toronto for learning English. I don't know if she remembered that, but she allowed me to go with the scholarship. And this was very important for me. So thanks so much.
And I was very picky. I didn't sign all papers. You know, a lot of students were mad at me because I didn't sign their papers. I say, no, you have to stay here.
So yeah,
So
actually,
when you went through a selection committee.
When I came with my plan, you told me, no, this is not a plan. You have nothing here. We have to go back home and do a plan and bring back to me again. So I brought the second version, and the second version was approved. So that's it. So thank you so much. Thanks so much, Rickson. Again, thanks so much, everyone. Ana, Mamadou, and also my friends, Romis too. Professor Romis, Andres, everyone contributed a lot for this. Thank you.
Congrats, Sergio. Excellent, well done, and all the best for you in the future. Lots of success.
Thank you for you too.
And please don't forget us. As mean people not in your group from Hickson, you'll be connected forever, I know. But, don't forget us, send news, tells us the news whenever you get some really nice result, tell us, we'll advertise here and so on. Okay. Don't lose your connection. Your connection includes our institute. So. Okay.
So thanks so much for all the kind words and everything.
You deserve more than that, Sergio. You did great. Right?
Thank you.
Hickson, do you plan anything else? Because I have a lot of people waiting for me.
No. I guess that's over now.
Okay.
Once I say the answer. Thank you very much, everyone.
Thank you so much.
Okay,
Thank you.
Hope to see you guys in person soon.
Yes.
Yes,
See
yes.
you soon.
Okay.
Take care.
Bye-bye.
Bye-bye, everyone.
Bye-bye.
Nice to meet you. Bye.
Bye-bye.